And we are recording.
Okay, thanks. So can I go ahead and start now?
No, we have to.
Let everybody in.
Yeah.
Okay.
I don't know if you're going
I've
to do it.
admitted everybody.
Yeah, please.
Everyone has been admitted.
Okay. So welcome everyone. I'm Alexandra de Sousa, and together with Tanya Calvé and Amélie Baudet, welcome to the From Fossils to Mind workshop. Henry Rollins said, every single record I have is a fossil. Fossils are records of the past. They give us natural history and dates by which to calibrate evolutionary events. It's with these fossil records that we can reconstruct the origins of species like our own. Marvin Minsky wrote Minds Are What Brains Do, providing a clear and simple understanding of how minds and brains relate to each other. One of the most common ways we understand what brains do is with functional neuroimaging. This figure shows activation of the Nile crocodile forebrain after binocular visual simulation. So therefore, if we want to understand the origin of any mind, including our own human mind, we're going to need some fossils. Form ever follows function, wrote Louis Sullivan. In vertebrate paleontology, fossils of hard tissues are used to understand the functions of different body parts. Structures relate to functions based on their physical properties in comparisons with other species. So we could, using just skeletal material that can be preserved in the fossil record, understand the locomotor behavior of these species. For example, we can see that the wombat does some burrowing, the horse runs, the lemur climbs, the seal swims, the elephant moves slowly, and humans walk on 2 feet. But when it comes to applying this principle to brain functional morphology from the fossil record, it's a lot more challenging. This is because, well, brains don't really preserve in the same way. And brain functional morphology requires examination at a smaller scale. We have to look at microanatomy. We have to look at molecular biology. And it's also really hard just to define the cognitive functions that the mind does. So the brain itself doesn't really become petrified in the way that bones and teeth do. The Tong endocast and natural endocasts like it, they look like brains, but they're actually molds of the internal cavity of the brain case, formed when the neurocranium is filled with sediment during fossilization. So endocasts provide some limited information about the size and the shape of the brain and some of the surface features. And artificial endocasts can be used in a similar way to understand these features. In addition to endocasts, we can get information, a record of the past, from other things as well. So archaeology and paleobiology provide information about material culture, tools, and other things that humans have made. Using them, we can understand behavior, and we can understand a bit about how the mind works. And actually, most of the research into the human mind's prehistory is done by archaeologists. We can also learn about ancient contexts from other kinds of paleobiological information. For example, ancient DNA can tell us about the genes that were present in species that are no longer around, such as Neanderthals. The comparative method can also be used to determine shared and derived aspects of neuroanatomy and cognition. Comparisons between species can be made at different levels, which correspond to the many scales at which brains are organized. So this is very important for drawing connections between the gross features that we can identify from endocasts and the microanatomy of brain circuitry And to understand how these differences arise between species in the first place, we need to look at studies of molecular biology and brain development as well. So the aims of today's workshop are to extract as much knowledge as we can from fossils about the minds of fossil hominins and other species. And so we'll be doing this by recognizing the context for which changes in brain size and organization occur, expanding neuroscience methods to study fossil endocrinia, and developing new approaches for understanding the brains and behaviors of fossils. So how will we do this? Well, we're providing a context for new approaches. We've invited 11 different speakers. We've got 6 ECRs and 5 students. And as a group, we'll be covering diverse fields, including anatomy, computer science, genomics, and biostats. We're going to be investigating the brain at diverse levels of organization, including molecular biology, cell biology, cytoarchitecture, connectomes, and the surface of the brain. The species we cover are diverse. We're going to be ranging from mice to dinosaurs. And we're going to be using all sorts of different interdisciplinary approaches as well. So our intended outcomes are to produce a review paper on the state of the art in understanding the minds of fossils that all of the invited speakers will be co-authors on. And we're also producing an edited volume, which will go into detail about the many approaches all in one place. Our other goals are to build long-term collaborations between this group of researchers and to create new opportunities and improve the opportunities for students, ECRs, and African young researchers. And finally, we want to make sure that this kind of research has impact and can be applied as well. So a little bit about the special volume and progress in brain research. We invite all speakers and ECRs to submit a chapter. We encourage collaboration between the speakers and the ECRs in authoring chapters. Send us the title by the 15th of December so we can forward this information to Elsevier. And the 3 organizers will be the ones who will be editing the volume. So an overview of what's going to take place over the next 2 days. So we're going to have 3 sessions of invited talks. There will be a public lecture today at the end of the day. We're going to have student and ECR flash talks. And we'll be brainstorming the paper in 2 sessions. So here's the schedule. So we start today with the welcome address, session 1, lunch break, session 2, coffee break, and the first brainstorming session about the paper, and finally a public lecture today. Tomorrow, we'll start with session 3, coffee break, the flash talks, paper brainstorming again, student prizes, and the closing address. So a bit more detail about the student and ECR presentations. So invited presenters will vote for student prizes. They must do this before 2 p.m. tomorrow. Check your email for the Google form that you will use to place your vote. So the presentations will, for student and ECRs together, will last approximately 1 hour and 30 minutes. So they're going to be 5-minute student talks and 10-minute ECR talks. You can watch them at your own convenience at the link here, and you should also receive that link in your email. And then tomorrow we're going to be doing flash talks by the students and ECR. So those will just be 2-minute talks, which will total 30 minutes. And this will be followed by a 1.5-hour Q&A session for the student and ECRs. At the end of the day today, we welcome Dr. Tawanai, who will be talking about digital data in museums, the creation, curation, and storage of it. So that'll be at 5 p.m. South Africa time, and you can join us through this Zoom link, which you should also receive in your email. And this will be open to the public, so please share with interested colleagues. So the Q&A sessions, if you're a student or an attendee, please enter your questions in the chat. If you're an invited speaker or an ECR, you may enter your questions in the chat, or you may use audio for asking questions. We'll be recording the whole meeting, but this recording will only be available to registered attendees. So we are the organizers. We are 3 scientists dedicated to understanding brain evolution. The 3 of us, Amelie, Tanya, and myself, form an interdisciplinary team. We're based in archaeology, psychology, and anatomy departments at different universities. Our interests encompass paleontology, comparative neuroscience, and also the application of evolutionary theory to translational research. And Tanya has been the Southern African Neuroscience Society chair since 2016. We'd like to thank the Southern African Neuroscience Society team for helping with the workshop. Rachel, Nanisi, Chris, and Brian. And we'd like to remind everybody that the Southern African Neuroscience Society is a nonprofit and thanks you for your support. Thank you to all our speakers, including invited speakers, ECRs, and student presenters. Thank you to all our sponsors for providing financial support. And finally, thank you to all of our attendees. So welcome. And I think we have a short break and then we'll be beginning the first session.
Alexandra, do you mind if I just make comments about the recording? If you feel you wouldn't like your talk recorded, what we can do is we could just edit it out of the final reel. So just let us know via email if you don't want your talk to be shared to the registered attendees. Anisi, should we maybe start playing the talks for the next 14 minutes or so?
Oh, I see an announcement you made in the chat here, Tanya, about the progress in brain research volume. So yeah, if you're a student and you would like to submit with a senior co-author, just let us know.
I don't think it's working now, the video.
Yeah, we can't hear.
Some global properties like interconnect.
Is there a problem with the videos?
Audio, but I think we started to hear now as soon as you unmuted yourself.
Okay, and then do you guys have the audio? The video, sorry.
The video, yeah.
Video, yeah, not the audio. Maybe just start again and unmute yourself.
Okay.
And
Can you guys see the screen?
yep.
And my name is Yongbing. I'm a researcher from the Dutch Connectum Lab at Free University Amsterdam.
And
It's
should
my
we
great pleasure today.
just to briefly say that this is the presentations from ECRs and students, so we're going to play them when we have time, like during breaks, etc. But you can also watch them using the link we sent. But it's just for you to have a chance to watch them like during the sessions. But yeah, and then so it means we will stop these videos by at 8. Sorry, 8. That's my time. By 10 when Julien will be the first speaker.
Here and present to my project about a brain-based parcellation method for interpreting brain organization from endocasts. You know that brain endocasts are very important for studying hominid brain evolution. It's simply because we don't have fossils for the real brain. We only have fossils from the brain case. So using the endocast, we can get a replica of the inner surface of the brain case. Therefore, we get indirect measurement of the real brain. We can easily extract some global properties like intracranial volume from the endocast. For example, we can design a study and extract the intracranial volume from human, chimpanzee, and the endocast of hominid fossils. And in this way, we can compare the intracranial volume, which can help us understand the evolution of the entire brain size. However, we also noticed that there are large regional variations in human brain evolution. For example, if we compare the human brain to the brain of chimpanzee, we can notice that the expansion of surface area in frontal cortex is much larger than the expansion happens in primary regions like occipital lobe. So we were always very interested in this kind of regional variations, which may help us understand the evolution of brain function. To capture those regional variations, we can use several methods. For example, we can draw some cerebral gyri and sulci according to impressions on the endocast, or we can also draw some landmarks according to the morphology or the shape of the endocast, and then make comparisons between endocasts using these landmarks. However, these methods are usually needs a lot of labor works and sometimes are also kind of subjective. So what we were thinking was that can we come up with another protocol which can help us easily study those regional variations on the endocast? We already know that in the field of neuroimaging using MRI, studying those regional properties, regional variations of brain morphology can be very easy. We can simply collect the data from ex vivo or in vivo human brain or the brain of other non-human primates, and then reconstruct the brain and parcellate the entire brain into different brain regions according to some brain atlases. So here, if we can apply those MRI-based brain atlases on the endocast surface, we may have the chance to study regional variations of the endocast. Therefore, we designed this study. We aim to first develop an MRI-based protocol to parcellate cortical regions in extant and fossil hominid endocasts. And second, we hope to estimate regional variations in surface area differences among endocasts of extant humans and chimpanzees, as well as fossil hominids. Here I show you the pipeline we made, which is used to first match the MRI data to the endocast surface, and then map the brain atlas at the MRI space to the endocast surface. And here I won't go into details about this pipeline. what I want to show you is that we tested this pipeline using several endocast samples, including one extant human individual, one extant chimpanzee individual, and one hominid fossil named STS5 from the Skerkfontein in South Africa. And this endocast was scanned using micro X-ray computed tomography. On the other side, we also used MRI data. We used two MRI data, one for human and one for chimpanzee. For human, we used MNI 152 atlas, which is the average MRI data from 152 human subjects. And for chimpanzee data, we used one MRI data from one chimpanzee individual. This MRI data were processed using free surfer to reconstruct the 3D brains and then parcellate cortex into different brain areas. And here we used two different atlases, including the Descan-Kiliani atlas, which includes 72 regions, and the Bonney and Bailey atlas of 76 brain regions. And here I'll show you some results of our mapping. First, you can see the Descan-Kiliani atlas on the human endocast. You know that the Descan-Kiliani atlas is defined according to the gyrification. So here we would hope the boundaries between regions would align to the sulcus of the brain. And here, this endocast is superimposed by the sulcal imprints. Therefore, these gray lines, we would expect them to align with the boundaries of the two regions. And you can see the alignment works okay, although there are slight shifts, but in general, it works very well. And here we mapped another atlas, the Bonney and Bailey atlas, on the human endocast. And this atlas was defined according to cytoarchitecture. Therefore, maybe this atlas may provide us the opportunity to study the changes, the regional properties of endocast in terms of the underlying cell structure. And besides the human brain, the human endocast, we also mapped the chimpanzee atlas to the chimpanzee endocast. Here we mapped the Bonney and Bailey chimpanzee atlas on the chimpanzee endocast. And in this atlas, we have homologous regions at the human Bonney and Bailey atlas. Therefore, it provides us opportunity to compare those regional properties between different species. But you know that the most important point of our study, of the endocast study, is to study the fossils. So we hope to apply those atlases on the hominid endocast. And here you can see the results after we applied the human Descan-Kiliani atlas on the hominid STS5 endocast. By visualization, it also works fair. And after this, what we want to do using this atlas is to make some comparisons between endocasts. First, we try to make comparisons between endocasts of human and chimpanzees to estimate the brain changes, the surface area changes, or we also call it brain expansion. And as you can see in panel A, using endocast of chimpanzee and human, you can see large expansion in prefrontal cortex and orbital parts of the inferior frontal cortex. And this pattern of expansion is very similar to the pattern we derived from the real MRI data. That is what we can observe using MRI in the real brain. So this like the high correlation between these two measurements indicates that using endocast, we can achieve a fair approximate of the real brain expansion. So in the next step, we make comparisons between the human endocast and the hominid endocast, as well as the chimpanzee endocast and STS5, the hominid endocast. Between human and hominid, we can see large expansion in the orbital part of inferior frontal, superior frontal, and lateral occipital regions. But between chimpanzee and hominid, we can see a slightly different pattern where the largest expansion was observed in rostral middle frontal, superior, and middle temporal regions. And in the next step, we hope to examine or discuss whether these different patterns would indicate different evolutions of brain functions. So in conclusion, in our study, using our protocol, we mapped MRI-based brain atlas to the endocast of extant human and chimpanzee and fossil hominid. And using endocast, we were able to estimate regional variations in brain area expansion among human, fossil hominid, and chimpanzee. Finally, I would like to thank my supervisor, Martin, and also my great collaborators, Emily and Alexandra. Thank you for your great support, and thank you for your attention.
I think we can go ahead with session 1. Are you on mute, Alexandra? No, we can't hear you.
Hello. Hi. So we're about to begin session 1. So we have 3 speakers in this session. And our first speaker is Julien Benoit.
Morning.
Good morning. So would you like to
Can I
have
share my
slides?
screen?
Yeah, share your screen.
Okay. Okay. Can you see my screen?
Yes, I see your PowerPoint.
Okay, cool.
Okay, it comes out perfect.
Okay. So whenever you want. Can I start?
Yes, you may begin.
Okay, thank you. So good morning, everyone. My name is Julien. I am a paleontologist at the Evolutionary Studies Institute of the University of the Witwatersrand. And today I'm going to give you a quick overview of how the mammalian brain got to be a special kind of brain. To do this, I'm going to focus on the brain, but also on the evolution of sense organs, because you would see that the evolution of the sense organs played a large part in turning the reptilian brain of our ancestors into the brain that we know today in mammals. So first, for those of you who are not paleontologists and who are not particularly therapsid paleontologists, the guys we are going to look at today are called the mammal-like reptiles or therapsids. And these animals are basically reptiles, and they gave birth to mammals. So mammals, to people like me, are just a weird kind of reptiles. And this is still kind of true today. This is evidenced today by the platypus, which is a mammal in every aspect, except that it lays eggs. So this is a remnant of our reptilian ancestry that we can still see today in the platypus. So here in South Africa, we are blessed with the Karoo, which is a vast semi-arid desert that covers most of the South African country. And the layers of the Karoo are very well exposed and document with great details the transition from reptilian-looking animals like this one, sprawling animals, into mammals as we kind of know them today. So small shrew-like animals that eventually gave birth to the mammalian diversity that we know today. So these layers here document that transition from reptile to mammals in great details. And today we are going to look at this transition, this very important part of their evolution, the transition from the cynodonts into the mammals. So cynodonts are the last reptilian-like ancestors of mammals. And what's interesting there is what basically turned these reptiles into mammals, which is an event we call the nocturnal bottleneck. So the nocturnal bottleneck is basically when those reptiles that dominated the terrestrial ecosystem turned into small shrew-like mammals when the dinosaurs basically became the dominant species on Earth. So our ancestors adapted to a nocturnal way of life. They became very small, lived in the night, and became insectivores. And that's how they survived the dinosaur time. So this is an oversimplified version of the story, of course, but for us, this is interesting because that nocturnal bottleneck basically was a sensory revolution for our ancestors. So because they became nocturnal, they lose the sense of sight, or the sense of sight was no longer, they did not lose it, of course, but it became less important, less crucial for survival. And they developed other sensory mechanism to compensate for the loss of sight, which is more accurate olfaction, more efficient hearing, and the sense of touch through the whiskers. So that's at this transition between cynodonts and mammals that we expect to see all this transformation in the brain. And to approach that transition, instead of giving you an overview of the 200 years that that we've been studying that transition, I'm just going to focus on the last 10 years, because 10 years ago, there was a statement by Timothy Rowe et al. In a very influential paper in Nature in 2011, so yeah, like exactly 10 years ago. And in this assessment, they summarized the knowledge, what we knew about cynodonts and their sensory adaptations and their brain with this sentence. So early cynodonts possessed low resolution olfaction, poor vision, insensitive hearing, coarse tactile sensitivity, and unrefined motor coordination. So they were super primitive, or at least so we believed in 2011. And since then, we've made lots of progress. So I say we, the whole community of paleoneurologists that who studied the therapsids, the mammal-like reptiles, and particularly the cynodont. So we've made lots of progress. This is basically the figure that Rowe et al. Put in their paper. This is in yellow, you have the total evidence that they have for the brains of cynodonts. And now you can see this is everything that we have today, 10 years later. We have cynodonts, but we also have so many other therapsids that have been documented. So the fossil record of the fossil brain, the endocast, as you presented, Alexandra, has, I don't know, grown 3 times bigger than what it used to be. And this is due, of course, to CT scanning. Now CT scanning has become more in, I mean, it's very well established in palaeoscience labs. And now, thanks to CT scanning, we have access to data that we did not have before. So here is an example. It's the sclerotic ring, which is the ring that goes around the eye and gives a direct access to the dimensions of the eye in extinct species. So thanks to that sclerotic ring, for example, Angelzik and Schmidt in 2014 documented the onset of that nocturnal bottleneck here in cynodonts. So that's one example just to illustrate the kind of techniques that have enabled this progress. So CT scanning and synchrotron scanning and neutron scanning were heavily involved, and with teams all over the world studying our South African fossils. So let's take those statements that Rowe et al. Made in 2011 one by one and see if they still hold true today in light of those new data. So first, the low resolution olfaction. So this statement was based on the fact that in mammals you have super big olfactory bulbs, whereas in cynodonts you have tiny olfactory bulbs. So that was very true at the time. Now that we have more data of those cynodonts, we realize that some cynodonts, such as Brasilitherium, actually had significantly big olfactory bulbs. In that particular case, in Brasilitherium, it's very striking because the olfactory bulbs are almost as big as the hemispheres of the cerebrum. So, and now we have a very nice transitional series where we can follow all the transformations of the brain in cynodonts up to the morphology that we see in mammalia forms. Morganucodon is known as one of the earliest mammalia forms for which the brain has been documented. So mammals start here, and here you have cynodonts where you see that transformation, the olfactory bulbs growing in size up to a very mammal-like size in Riograndia and Brasilitherium, so before the origin of mammals. So we are a couple of million years before the origin of mammals. So did all cynodonts had a low olfactory resolution? Probably not. Some of them were already very mammal-like before the onset of mammals. Poor vision now. Another statement was that our cynodont ancestors had poor vision. So this is probably the statement that's the most wrong, especially in light of new genetic data and new molecular data that basically demonstrated that the poor sense of sight in modern mammals, and particularly the color vision. Most mammals are bichromatic, and actually what they show is that going back in time, our ancestors, our cynodont ancestors here at the base of the tree, did not have all the mutations that turned the tetrachromatic ancestral condition into the bichromatic condition. So that means that our cynodont ancestors likely was tetrachromatic, could see, well, had 4 different types of receptors for colors, while the color vision was lost probably convergently in monotremes and in eutherian mammals. So our ancestors had a much better color vision that we had, and that of course is due to the nocturnal bottleneck, because color vision was no longer a condition for survival during the nocturnal bottleneck. Now, insensitive hearing. So hearing in therapsids, at least as far as we can reconstruct, was limited to low frequency sounds in cynodonts. And this is probably because they had a sense of hearing that was basically reptilian, which is they were sprawling animals, so their head was very close to the ground, so they could listen for the vibrations of the ground, which are low frequencies. So that's what is illustrated here. But with the onset of parasagittal posture, with erected limbs, the head moved away from the ground. So with the mammalian posture, the head moved away from the ground, and so they had to adapt to higher frequency sound, airborne sounds, instead of seismic sounds, seismic vibrations. And this transition was not sharp. It was quite smooth with the development of what is called the cochlear recess. So the cochlear recess, which is that triangular cavity here, developed into a finger-like process that eventually curved and coiled into the modern mammalian cochlea. And this is a very interesting phenomenon, very well documented phenomenon. And now we have a complete series of transformation of that cochlear recess into the mammalian cochlea, with the first stages of that transformation happening already in cynodonts. So even though the cochlear recess of the most derived cynodont, which is here Tritylodon, is still straight and not yet curved like in the platypus, that cochlear recess is already well developed and probably enables some higher frequency hearing than the more basal cynodont. So same thing, it's a transformation that started before the onset of mammals, before the origin of mammals. And this is not the only specialization that we see in the hearing system of cynodonts. So here I'm showing you a fossorial snake here compared to a regular snake. And you can see that the vestibule, so the equivalent of the cochlea in snakes, is much more inflated than a regular vestibule. And it's because that inflation is an adaptation to super low frequency hearing. It's basically, it's the same thing you see in moles, for example. So to hear very, very low frequencies, seismic waves, to be more sensitive to seismic waves. And we see that kind of adaptation in some cynodont, for example, cystecynodon. So cystecynodon is very interesting because it does have those very inflated vestibule. So the cochlear recess turned into that big inflated vestibule. And in the meantime, the pineal foramen here for the pineal eye shrinked in size and become relictual, which is also something that you would interpret as a fossorial adaptation, because when you live underground, you don't need, well, a reptile that lives underground doesn't need a pineal foramen anymore. The pineal foramen can only be used if it's exposed to sunlight. So overall, cystecynodon sends the paleoneurology of that cystecynodon give us a nice consistent signal that this animal was adapted to a fossorial way of life, and so developed its own specialization, its own sensory specialization that are very different from those of modern mammals, but definitely not an insensitive hearing. The coarse tactile sensitivity now. So the statements by Rowe et al. About coarse tactile sensitivity was mostly based on the fact that mammals do have whiskers, and that these whiskers allow whisking, which is a tactile movement that sends it sends tactile signal to the brain. So this is definitely a high. This enables a high tactile sensitivity. This tactile sensitivity is transferred to the brain through the trigeminal nerve that goes through the infraorbital foramen. So it is usually accepted that the infraorbital foramen is the osseous correlate to the presence of whiskers. That can be debated, of course, but as I say, I'm giving you a simplified version of the story. Long story short, when you look at our mammal-like reptiles, one can only agree with Rowe et al.'s statement that our mammal-like reptiles probably did not have whiskers, judging by this very crocodile-like patterning of the maxilla. That is definitely indicates a crocodile-like skin on the face and not whiskers. And there's no, as you can see, there is no infraorbital foramen in our mammal-like reptiles. So now with, again, CT scanning, we could CT scan a larger sample of therapsids and document the transition from the reptilian-like type of trigeminal nerve into the more mammal-like type of trigeminal nerve. So as you move towards more derived animals, so from top to from top to bottom, you're going from more primitive animals to more derived animals at the bottom. So the earliest, most reptilian-like pedicosaurs had a very complex, very highly branching trigeminal nerve, completely encased in the maxilla. So no chance of whisking in those animals. And as you move up into cynodonts, and particularly the most derived cynodonts, you see that the canal in the maxilla becomes more and more simple. It has less and less branches, and it's not because the nerve becomes simple, it's because the nerve is no longer encased in the maxilla. So the nerve now ramifies in the flesh, which enable whisking movement. And following the transformation from a nerve encased in the maxilla into a nerve free to move in the flesh and no longer encased, we could pinpoint where the evolution of whiskers was the most likely to occur, and it's probably 241 million years ago in those derived cynodonts, which have an essentially mammal-like infraorbital foramen. So again, another feature that evolves, another mammalian features that evolves prior to the mammalian origin in our cynodonts. So not all cynodonts had coarse tactile sensitivity. And this was confirmed by Myszlinski et al. In 2020, who plotted whisking mammals and non-whisking mammals based on the surface area of their infraorbital foramen, on the dimensions of their infraorbital foramen, and found our cynodonts, our derived cynodonts, here named Pachygenus, among the whisking animals. Among whisking mammals. So definitely not all cynodonts had a coarse tactile sensitivity. And finally, the unrefined motor coordination. So of course, it's difficult to quantify motor coordination in an extinct animals, but looking at the floccular fossa, so for the floccular lobe, which has function into stabilizing the gaze, and looking at the evolution of the cerebellum in general, we see that the floccular fossa was extremely well developed in our cynodonts. Actually, most mammals have a smaller floccular fossa than our cynodonts. And the cerebellum, so the cerebellum develops as no vermis in the more primitive cynodonts, but again, Rio Grandia and Brasilitherium, these two again, developed a vermis of their own on their cerebellum, and their cerebellum is also broader, which also could be somehow indicative of a better motor coordination. So again, motor coordination is not just mammals. Some of the most derived cynodonts were already essentially mammal-like in this respect. So overall, the evolution of the brain in mammal-like reptiles is very interesting. So if you follow brain size, here you can definitely separate placental mammals and non-avian reptiles and amphibians on that plot of body mass against endocranial volume. But then what's interesting in that plot is when you add up marsupials and monotremes, and then if you add up again Mesozoic mammals, then you see you get progressively closer to the non-avian reptiles. And then if you add our cynodonts to the mix, then the cynodonts give you a very nice transitional buffer between your reptiles and your placentals. And what's even more interesting is when you consider phylogeny on top of that, the most derived cynodonts are located right here. So again, Brasilitherium, Rio Grandia, always the same. They are located right here, right at the transition, you see, between reptiles and mammals, with basically the place where reptiles and mammals overlap. So there is a very nice signal also in the brain evolution that tells us that not all mammals were as derived as we used to think, and not all cynodonts were as primitive as we used to think 10 years ago. So overall, cynodonts for many sensory features were no worse than early mammals. Early mammals were actually, as I said, kind of primitive, and derived cynodonts were as good as them in many respects in terms of sensory abilities. And some cynodonts were even very derived in their own rights, like cystecynodon, for example. And for the vision was color vision was much better actually, probably in cynodonts than in mammals. So the nocturnal bottleneck. So now to summarize really the onset of the origin of the mammalian brain, I will use the model developed by Rodriguez et al. In 2018. So it all comes down to the nocturnal bottleneck. So because of the nocturnal bottleneck in the early Mesozoic, dinosaur rises, mammals shrink in size, become nocturnal, which changes the sensory input in their brain. So that's what represented right here. The sensory input to the brain changes dramatically in the early Mesozoic. So because of the small body size, the interoral distance is much smaller and the vocal cords are much smaller, so they cannot hear or produce low frequency sounds. Also, because of the parasagittal posture, they cannot use seismic waves anymore. So high frequency hearing becomes important. And because they lose color vision, they have to compensate with smell, hearing, and touch. So with whiskers, large olfactory bulbs, and a cochlea. So that changes in a second step, that changes their receptors. So they develop the cochlear recess into a cochlea. They develop large olfactory bulbs. They developed an infraorbital foramen that enables whisking, and they lose the pineal eye, which is a reptilian feature that gets the sunlight. And finally, the brain responds to those changes in the receptors because of the new somatosensory input. The somatosensory cortex grows, which helps the brain to reach an higher encephalization quotient. The cerebellar vermis also develops, and the colliculi develops, all of this contributing to an overall increase in brain size. So that's where we stand today in terms of explaining the origin of the mammalian brain, so that enlarged brain that originated in the early Mesozoic, and that formed the basis of all the crazy developments in brain and sensory evolution that we will I'm sure we are going to cover later during this workshop. Thank you.
Thank you. Thank you for a fascinating talk on a really fast-moving field. So we have a few minutes left, break to change over to the next speaker. We'll take the questions at the end.
Okay.
So good morning, everybody. I'm Pascaline Lauters. I did my PhD at the Université Libre de Bruxelles and Museum of Natural Sciences on the general structure and evolution of the brain of ornithischian dinosaur, and more especially ornithopod dinosaur. I'm going to start by the quick introduction. So about dinosaurs, they were a very diverse group. They can be separated first into the Saurischian dinosaur, who are composed of the sauropodomorph. Everybody knows the brachiosaur, the diplodocus. They were characterized by usually animals of very big size, long neck, long tail. Also the theropod dinosaur, the most famous ones that are most known for the carnivorous dinosaur, but actually a lot of them re-evolve a herbivorous diet. They're the only dinosaur that we observe today because birds actually come from that lineage of dinosaurs. and the birds are the only dinosaurs that went through the Cretaceous Paleogene extinction. And the other group is the ornithischian dinosaurs, where herbivorous dinosaurs who were really diverse. You have the stegosaurus and ankylosaurus. You have the triceratops and pachycephalosaurs. And my work has been focused on the ornithopod dinosaurs. They were very present since the Middle Jurassic until the end of the Cretaceous. Very big groups. They were present everywhere on the continents. And we're going to see today what happened or didn't happen in the development of their brain. So there is a problem when we study, of course, we want to study the brain of extinct animal. It's at first we can only have access to some preservation where really in exceptional circumstances you can have mineralization and fossilization of the brain, but it's very seldom. And sometimes we could find natural ondocast that were preserved, but it's something that was rather uncommon. Sometimes we couldn't assess who was it was sometimes very difficult to identify the dinosaur to whom belonged the natural ondocast. So to expand the data sets, paleontologists started to make casts at first in plaster. So they will fill the endocranial cavity with plaster, and then we'll have to break or cut open the skull to retrieve the cast. So it was a very destructive method. It will at least damage and sometimes even destroy the specimen. This was not such a nice way to do it. Fortunately, we now have other methods, and we will use materials like silicone, for example, with artificial material with elastic. It's possible to make a coating of silicone inside the endocranial cast and then retrieve it. And it will give us a lot of details without damaging the skull. So it's a very interesting method to use. And another method that's been widely used in past years is a CT scan. This is also a very nice method. It allows us to once again expand our data set because some specimens for taxonomic reason, for preparation reason, because they're very fragile, cannot be molded. We cannot make casts of the endocrinal cavity, and using CT scans to compute tomography allow us to still make reconstruction of the endocrinal cavity of this specimen. This is a small, if it works, yeah, a small example of the skull of an iguanodon. One of the iguanodon I was able to scan and then to reconstruct the endocrinal cavity. So the objective of my work were to see if the anatomy of the brain was stable in the ornithopod lineage, and if we could possibly make some inference about the ecology and the behavior of this group of dinosaurs. I want also first to have a small talk, I would say, about something that has been pervading the literature, and you probably often heard, and something that was very present in books some years ago still, the fact that a dinosaur had a small brain. This is coming from one of the very most famous fact you will find in books is that Stegosaurus, who was a rather big late Jurassic dinosaur, yes, you can see it had a rather small head, so the animal will be more than 6.5 meters long, weight more than 4 tons, but the brain of the animal will only be the size of a walnut. And people have been saying that, jumping to the conclusion that because the brain was small, they had to be not so intelligent. And I want to talk about that because there was also an idea that we still found a lot in literature. So for birds and mammals, the brain completely fill up the endocrinal cavity. And so when you have a cast of the endocrinal cavity, you have a really good proxy to know the size, the volume, and to distinguish the feature of the brain, of the real brain of the animal. This is not so much the case for some reptiles, and it has been noted that there is sometimes a lot of tissue between the surface of the brain and actually the bones of the endocrinal of the endocranium, and this led to estimation that for dinosaurs and some extinct animals, when you look at the endocrinal cavity, the brain itself of the animal will only fill 50% of the endocrinal cavity. So you have already a big animal, a small head, a small endocrinal cavity, and then they will still shrink the approximation of the brain of 50%, because it's something you will see in crocodiles. But actually, there is a lot of variation between species, and even inside a species, I don't know any rule about that. I wanted to introduce this because actually in dinosaurs, what we can find is valleculae. So valleculae is the term coined for the prints of blood vessel on the endocranial roof. And this is really interesting because it's something that will only appear when the brain comes closer in contact with the bone, when there is not so much other tissue in between the surface of the brain and the bones itself, so that the blood vessel will come mark the bone actually. And this is something that has been observed first in some North American hadrosaur and pachycephalosaurs. And it tells us that at least for this part of the endocrinal cavity, the volume, the surface, the feature we can observe more certainly reflect the brain of the living animal. And it's something I've also been able to observe on some Asian or ornithopods. So finding it is another example. You see the black arrow showing the valleculae. This is really interesting because it tells us that part of the brain is more precise, and also the brain was filling more of the endocrinal cavity. And we usually then use a proxy of the brain will fill up more than 60% of the endocrinal cavity for these animals. So I will now proceed to show you a few examples of the brain of ornithopod dinosaur. First, two iguanodons. On the left, Iguanodon bernisartensis, as you can see, has a rather straight endocast, rather large cerebral hemispheres, and rather large olfactory tracts. Was not able to reconstruct the olfactory bulbs. And I want to, so the picture on the right is one of Mantellisaurus, another iguanodon. A much more gracile form. Unfortunately, the specimen I could scan, the skull was looking in very good shape, but the inside had been crushed, and I was not able to make a full reconstruction of the endocrinal cavity. But I want to show this because we still see the cerebral hemispheres rather large, but the shape of the brain is really different, and we have a kind of S shape, a flexure, which is really interesting, and I will come back to this later, but it's something we can observe in other group of dinosaurs, or the brain cavity is not so straight, but actually you have angles between the different parts of the brain. The next specimen is Lourdosaurus. It's also a big iguanodon from the early Cretaceous from North Africa. Once again, the endocranial cavity is very straightforward, but we see some other interesting features. The olfactory tract are very small, actually. This is very small compared to iguanodon. the cerebellum, the cerebral hemispheres are not so much largely expanded. They're more in the more oval shape. And I was baffled by the pituitary fossa, which is really small, which is strange because this is a very big animal. So I'm not so sure how to interpret this, but maybe it could be an artifact of preservation. But for big animal, it should have a big pituitary fossa, and this one is rather small. This is the endocrine cast of Batyrosaurus. This is an animal who's between the iguanodons and the hadrosaurs or duck-billed dinosaurs. It is coming from Kazakhstan, early Cretaceous. And this is, again, you see a more straight endocast, a large cerebral hemisphere. This is you don't see it so much on this picture, but there are valleculae, and this is to this point the oldest occurrence of valleculae in this group of dinosaurs. So with this part of the brain, we can say we have a really good picture of what it looked like, very much expanded. Unfortunately, we're missing the frontal part of the endocast. And it was a very good specimen, and I had also a cast of the semicircular canal. I want to say a word about this. I didn't delve too much into it, but seeing the proportion of the semicircular canal, it will indicate that this animal was probably a biped. No, this is something we would like to confirm with more data, but we don't have many bones at the moment, so we cannot make a complete reconstruction of the animal, but it will show an animal. We are expecting an animal that was more gracile and probably more bipedal than other members of the group who are usually quadrupedal. I then go forward into the ornithopods and reach this is an example of the duck-billed dinosaurs, the Amburosaurus, highly derived hadrosaur from Russia, from late Cretaceous. We had a lot of specimens, but a lot of them were incomplete. And so this is the undercast of one of the beds skull, and you see once again a very straight endocranial cavity, really large cerebral hemispheres, very expanded, and very large olfactory tracts. And here you can see pituitary fossa is more expanded. That's what we could see in Lourdosaurus. So what can we maybe say about the sensory organs and the variation in this group of dinosaurs? Most of them seems to have big olfactory tracts, and so probably a very good sense of smell, except Lourdosaurus, but other ones, iguanodons and duck-billed dinosaurs had large olfactory tracts and a good sense of smell. I'm coming back to the flexure because it's something very interesting that has been published also by my colleagues Lautenschlager and Hübner, that flexure are actually could be, how would I say, there is, it could be a mark of the room available for the brain to expand. I mean, in smaller animal, when the brain doesn't have so much room to expand, because the skull is smaller, you will have flexure, and it's something I did some cast on birds or extant birds, and it's very impressive to see how the brain is really packed. And because it's kind of pushed back by the eyes, the orbits are really big and pushing back the brains. It doesn't have much space to expand. and the brain will be really packed, and the flexure will be really important. What Lauters, Schlager, and Hübner did, they were able to study an ontogenic variation in Diceratosaurus, was a Middle Jurassic ornithopod dinosaur, Iguanodon dinosaur from Tanzania, where they had young individuals and more adult, more mature individuals. And you can see that in the same species, the shape of the brain will actually change with ontogeny. That in the small animals, the flexure is more pronounced than in larger animals. The flexure is not so much pronounced. Flexure are not so present so in ornithopod dinosaur, except in Mantellisaurus, was not so surprisingly smaller, more gracile form. But in iguanodons, other iguanodons and hadrosaur dinosaur, there is the flexure basically disappear and you have a straight undercast. I also did some evaluation of the encephalization coefficient. So, of course, when you have a very small animal, it has comparatively a very small brain compared to a big animal. And a way to compare animals of very different size, it will be to plot the volume of the brain compared to the volume of the body. I've been using the equation of Hilbert, was more especially designed for reptile. And as you see on the right of my slide, this is some data coming both from the literature and some of my work. So an EQ of 1 indicates that the brain is exactly the size expected for the size of the brain is exactly the size expected for the size of the animal. And so if it's below 1, the brain is smaller than what you will expect for the bees. And if it's higher than 1, the animal has a comparatively bigger brain than expected. So you have here the ostrich, who has a rather big brain, 4.96. So the ostrich has a big brain compared to its body size. You have here some theropod dinosaurs. You have some variation because of the uncertainty with reconstruct the volume of the brain, knowing the body mass. There are different ways to evaluate the body mass of the dinosaurs. But globally, you had rather high-brained animals, the biggest one being Troodon. This is actually the same estimation than for humans. We have an EQ going around 7 for Homo sapiens. You have here 2 sauropod dinosaurs. So these dinosaurs were very, very big, among the biggest dinosaurs, but comparatively small heads. And so you have also an EQ reflecting this with being sometimes rather low EQ, some of the ornithischian dinosaur. And here we have the ornithopod dinosaurs. So you see for ornithopod dinosaurs, all the numbers are above 1. So these dinosaurs are actually a rather bigger brain than what would be expected for their size. And I want to talk more about the cerebral hemispheres. I found this very interesting because this is the part of the brain that is most related to high cognitive function, like memory, socialization, complex behavior. And this is a part of the brain that seems really expanded in the ornithopod lineage. And there is something, it's a group that's very diverse, but also very well known. And we've found bone beds of ornithopod dinosaurs, hadrosaurs here. This is usually a clue that these animals were living in very large groups. They were living together, so they probably had a kind of communication, of socialization. This is a drawing of a nesting ground of Maiasaura, a dinosaur discovered by John Horner, and very famous because we can be pretty confident that this dinosaur were actually building next together, a tiny bit like seabirds who will gather all at the same time on an island and all nest together. This is what we can observe with these dinosaurs. So they were nesting on the same spot, maybe several times, I mean, always the same spot, all together at the same time. There has been hinted there was parental care because the baby would need some protection, or at least some form of care from the adults, which once again is something that is sure rather cognitive development. And there was more and more dinosaurs can be described of animals that were really active, some that's very social. We have proved they had sexual dimorphism, and they would engage into parades, and most especially for hadrosaurs who had crests and able to communicate through sound. There will be some kind of communication of high communication between them. And that will make sense with the enlargement of the cerebral hemispheres that will allow also to this process of behavioral complexities to take place in this group. Thank you for attention.
Thank you very much, Pascaline, for a really fascinating talk. I learned a lot of things about dinosaur brains. We have a few minutes, but we're
So can you see my screen?
Yes, it's perfect. You may begin.
Great. So my name is Felix Stroeckens. I'm working at the C&O Vogt Institute of Brain Research in Düsseldorf, Germany. And in my research, I'm focusing on the neuronal foundations of higher cognitive functions, which is also the topic of my talk. And my talk will actually be a little bit different in comparison to the other talks, because I'm not working with fossils, but I'm also interested in the evolution of brains. And in my research, I'm focusing on species which are in, let's say, interesting phylogenetic positions. And therefore, I hope that my research data is also interesting to the other people. So let's start. A lot of people ask me, if you're interested in higher cognitive functions, why are you actually focusing on sauropsid or avian species? Shouldn't you be focusing more on mammalian species, because those are normally the species known for well-developed cognitive functions, especially primates. But as most of you probably know, over the last recent years, a lot of studies could actually show that also species outside the mammalian clade have some very interesting cognitive functions. And they're mostly avian species like parrots or corvids show fascinating cognitive abilities. For example, we can find the cognitive ability to use tools or to construct tools in corvids and also parrot species. We have other examples. For example, there's a lot of data on cooperation in avian species. We have evidence for causal reasoning. For example, in New Caledonian crows, we know that they can make use of the physical properties of objects. And other examples would be self-control. Ravens show amazing self-control capabilities. They can wait longer for a reward than non-human primates and even human children can. Other examples comprise episodic-like memory in jay, for example, and there are even some researchers suggesting that corvid species might even possess something like a theory of mind, which was thought for a long time really be a domain of primate species. And these cognitive abilities are very interesting from an evolutionary point of view. And the reason for that is that mammals and sauropsids on the other side, here shown in this very nice graphic of Stefan Reber, are separated by approximately 300 million years of divergent evolution. And that makes it very likely that these cognitive abilities developed or evolved convergently from each other. And then it's on its own is already pretty interesting, I would say, but it gets even more interesting if you have a look at the brain layout and brain structure of these two different species group. And I do not want to go into detail here because I think many people know that, and we likely will hear more about that. But the brains of mammals and sauropsids or avians are very, very differently organized. In mammalian species, we have a telencephalic or a pallial organization, which is layered in form of the neocortex. So a very nice 6-layered structure. And in birds or in sauropsids, we more or less lack a cortical organization. Obviously, I'm aware that in several reptilian species, we find something cortical-like, but it is not one-to-one comparable to the neocortex in mammalian species. Instead, the pallium is organized in a nuclear organization, so we do not find like a specific layering. There are some recent findings showing that, well, we have at least the cortical-like connectivity, and also genetic fingerprints of cells in the pallium of birds is relatively similar to cells in specific cortical layers, but in general, we still have a big difference. And the main question of my research work is how can these very different brains can come up with comparable cognitive capabilities? And I'm focusing there on a couple of different factors which likely play a role for the development of cognitive abilities, not only in avian species, but also mammalian species. And these factors are, for example, things like the general brain layout, the layout, and also the size of specific, and there especially associative areas. Neuron numbers likely play a very important role, the number of neurons in the whole brain and also in specific areas, the connectivity between areas. The metabolism is very important, and I'm going to talk about that a little bit more on the next slides. And also synaptic transmission is there obviously very important. And there are many, many further factors which likely play a role. But in my research, I'm focusing on these factors. And today I would talk about these two things. This is first the layout of areas and the metabolism. And I would like to present you today a couple of studies in 2 specimens I'm analyzing, or I have analyzed in the past. This is, are the Nile crocodiles on the one hand, and there I would like to show you the results of an MRI and fMRI study. And second, I would like to talk about a PET study in pigeons, but more about that later. So let's talk about MRI and fMRI studies in Nile crocodiles. And there the question again is, why are you analyzing crocodiles, Felix, when you are working with avian species normally? And the reason for that is the phylogenetic position of crocodiles. Crocodiles form together with avian species the group of archosaurs, and they share a stem amniote ancestor, or an ancestor, and they are therefore the closest extant relative of modern bird species. And that makes them on their own already interesting. Furthermore, crocodilians changed their phenotype relatively little over the course of evolution. So they are kind of an ancient species, at least for a person like me working on modern species. And they are thus, for us, an ideal subject to study the ancestry of avian brain evolution. But unfortunately, there's very little data on crocodilian brain structures and brain functions. There are some beautiful histological studies and also several functional studies. But if you compare that to the amount of studies we have in mammalian or avian species, there's almost nothing. So we decided a couple of years ago to fill this gap at least a little by performing 2 studies in Nile crocodiles. And with we, I mean my colleagues Xavier Lü and Mehdi Behrozi, and most importantly, Brendan Billings, which is actually situated in Johannesburg. And we decided to do 2 studies. And the first study was a basic MRI atlas study in combination with histological analysis, where we try to identify different brain areas in the crocodile brain in adult post-mortem Nile crocodiles. In the second study, we planned and we also did a fMRI study in juvenile Nile crocodile, like for example this guy here. This is Lars, one of our specimen, and we got some really good data for him. So I'm also very thankful to Lars for letting us scan his brain. Very briefly, I would like to talk about the first study. I'm not going into much detail here, also not from the technical side. I would basically only tell you a little bit about the things we found here. And what we did was we put the animals in, or the heads of the animals in the scanner. Those were, as I said, post-mortem specimen, and we did long-term MRI scans, then sliced the brain, stained them with several histological techniques like Lissel or silver myelin stain. And then we used this data to create a three-dimensional atlas of the Nile crocodile brain. And we found approximately 50 areas. We modeled them in 3D. And if you're interested, this data is available on the homepage of Brain Structure and Function. And what is also interesting, that there was a relatively high concordance in the layout and also the localization of these areas in comparison to birds. So at some point in the future, we might need to think about a unification of nomenclature between reptilian and avian species, but that is definitely something for the future, but I'm happy to also discuss this later with you. Probably more interesting are the results of the fMRI study. Here we made use of several juvenile crocodiles, and the reason why we used juveniles was we couldn't fit bigger animals in the scanner. So our scanner setup was a small animal 7T scanner, and we only could fit juveniles in there. And we had to make a lot of custom equipment because the commercial available equipment is normally for rats and mice and sometimes for avian species. And it is shown here in this picture. So we had a custom made animal holder. We placed fiber optics in front of the animal eyes for visual stimulation. We had loudspeakers placed on close to the ears, and we used a surface coil for the recording. And possibly more interesting, what kind of stimulation did we do? We decided to do a visual stimulation in a simple on-off block design, and we used green and red light at different flicker frequencies. But this is nothing special. This is like a classic stimulation protocol used also in mammalian and avian species. And then we also did an auditory stimulation. There we used two classes of stimuli, simple stimuli, which were random chord stimulus centered around 1000 and 5000 hertz. And we also used here a complex stimulus, which was the first 12 seconds of the Brandenburg Concerto No. 4 of Johann Sebastian Bach. You might ask yourself now, why are you playing classic music to a crocodile in a scanner? The reason for that is because it has been used in the past for other species as well, because classical music has a wide range of different acoustic features, and it's therefore ideally suited to activate higher associative, not higher associative, but higher auditory areas. And it has been used, for example, in songbirds, and there it could activate a couple of areas. And therefore you can get very interesting data with that. In the very beginning, we also planned to do somatosensory stimulation and olfactory stimulation, but we ran into quite a lot of problems. And since some of you in the future might want to do fMRI experiments in other reptilian species, I would like to briefly talk about that as well. Because also these things are normally not mentioned in the papers. So I think this is something interesting to talk about. For the first problem, almost killed our whole experiment, and that was an anesthesia problem. If you are working with fMRI, you probably know that you cannot use all anesthetics to sedate the animals because this has an effect on bolt signal. And therefore, specific anesthetic, medetomidine, is used. An adrenergic agonist, not that important, and it works really good in rodents and also in avian species. It almost didn't work in crocodiles. It only caused a very mild sedation, but no immobilization. And that meant that the animals, although they were restricted in their carrier, they still could move. And if you move an animal with very strong leg and tail muscles into a very expensive piece of equipment, well, that is frightening. But for us, very fortunately, the crocodilians apparently liked the scanner situation because they didn't really move. Honestly, they didn't move at all. And this is shown here. This is basically showing the movement of the animal in X, Y, Z directions. And the scale is in millimeters. they basically didn't move. Sometimes they slightly moved their head and we could cut that out easily, but in general, there were no movement, and they were holding still during the hours we were scanning them, and that was very, very fortunate for us. The second problem is more of a technical problem, but also interesting, especially given the fact that reptilian species, including crocodiles, are poikilothermic species, and that might explain some of the problems. The bolt signal in these species is relatively low in comparison to avian and mammalian species, with a bolt signal change of only 0.7% in response to the stimulation. And also it takes longer until the bolt signal peaks. And it took us actually a week to figure that out and to adjust our protocols. And furthermore, the breathing has quite a big effect on the bolt signal as well. That is shown in this graphic down here. This is a response of the bolt signal in response to the stimulus. And you can see when there's a stimulus onset, the bolt signal goes up, or when there's no stimulus, the bolt signal goes down, but it goes especially up when the animal is breathing. And crocodiles have this very deep breathing bouts every couple of minutes. And this basically then prevents you to record anything. And therefore you later have to analyze the data and cut that out. But importantly here, you need to adjust the protocols. You cannot just use the avian or mammalian protocols. So much about technical problems. Let's go on with some results. And here's the result of the visual stimulation. And irrespective if we used green or red light, and also irrespective of the frequency, we always could activate or found activation in the nucleus rotundus here and in an area we called CVS1 in the telencephalon. And this is very, very comparable in layout and localization to the avian entopallium. So it's very likely the homologue of the avian primary visual area. Obviously, we cannot finally say this because we need additional, for example, genetic data on that, but it's very likely that this is really the homologue to the avian primary visual system. Here are the results of the auditory stimulation, and here we could find 3 different areas which were activated by our stimuli. And the first areas we called CAU1 and CAU2 were activated by all stimuli. And again, so they're shown here, CAU2 and CAU1, and based on their positioning, it's again very likely that they are homologues to the avian counterparts, namely the field L and the Nidopallium intermedio medialis, which are known to be primary auditory areas in birds. The third area was very interesting because it only was activated in response to the complex stimulus, so the Bach music. And this is interesting because it is known from avian species that these complex sounds only activate higher auditory areas, namely the Nidopallium caudomediale. And we think that this area we found here is likely, again, the homologue to the Nidopallium caudomediale. But beside that, that's also in another view interesting because it also shows that apparently reptilian, or at least crocodilian species, process auditory information in a comparable hierarchical process. And with this, I mean, the more complex stimulus, in this case an auditory stimulus, gets, the more and more areas are recruited. And we find the same in avian species and also in mammalian species. So it looks like this organization principle is already or was already present in crocodilians, and therefore already present or likely present 240 million years ago. To sum the study up, and with this the first part of my talk, I hopefully could show you that MRI and fMRI are in general possible in reptilian species, and that a one-to-one transfer of mammalian or avian fMRI protocols is not possible. I could also show you that we have comparable primary auditory and visual areas in crocodiles comparable to birds, and that we have a comparable hierarchical processing of sensory information within crocodilian species. I would like now to go on with something rather different, which is still an imaging study, but in this case in a different species, in a pigeon. and also some kind of different imaging. And in this case, we were working with positron emission tomography, and we did that to analyze the energy consumption of the pigeon brain. And again, that would be a great point to ask the question, why did you do that? And the reason for that were findings on the brain structure or brain composition of bird species in the last years. In 2016, a study from the Czech Republic figured out that avian species, and they are especially songbirds and parrots, accumulate much more neurons in their relatively tiny brains in comparison to mammals. If you, for example, compare the brain, rather big brain for a bird of the sulfur-crested cockatoo with that of a similar sized primate, the galago, you see that the brain weight approximately is the same. But the neuron numbers in the cockatoo are more than double as high as in the primate species. And that could be one explaining factor why birds with their relatively small brains in comparison to extant mammalian species can perform, or at least some of them can perform on such high cognitive levels. And I actually did a follow-up study on that where I analyzed the neuronal composition of brains of different avian species. And what I could find was that especially corvid species accumulate a very high number of these already high neuron numbers, especially in their associative areas, the meso and nidopallium. That is interesting, but it also brings up another relatively big problem. And the problem is the following. Brain tissue is extremely expensive energy-wise. For example, the human brain takes about 2% of the body mass, has 86 billion neurons, but it consumes 20% of the total energy intake. In rats, it is 0.6% of the body mass, 189 million neurons, but it still requires 5% of the total energy intake per day. In pigeons, we have a comparable brain size in relation to body mass, but the neuron number is almost double as high as in rats, and we currently do not know anything about the energy consumption in pigeons. And the question there is, how can pigeons and even more other avian species with very high neuron numbers can afford so many neurons? And my former PhD student Kaya van Eugen and two colleagues from Cologne University wanted to figure that out, and we did that by PET scans. We plan to do that in 4 different avian species, but today I will only talk about pigeons. And that, what did we do with these pigeons? We injected them with radioactively labeled fluorodeoxyglucose, or FDG. FDG is a glucose analog, which gets taken up like glucose by any cells in the body, but it doesn't get metabolized. So it accumulates in the cells and therefore can be measured and can serve as a proxy for glucose consumption and therefore energy consumption. And afterwards, we put the animals in the scanner, scanned them. We did that in a lot of different control conditions. We did that in awake animals, anesthetized animals. We did different kinds of blood sampling, and that was very important. And you will see on the next slide why, or on the second slide why. But today I want only to focus on awake animals and a little bit on anesthetized animals after IV injections. And the results are here. And the first picture basically shows that the PET scan was working. We have here the MRI picture of the pigeon head, and here we have the overlay of the PET scan, and you can see that the brain was taking up radioactively labeled FDG, and not only the brain, but also the retina was taking up quite a lot of FDG, which is relatively well known from the literature and other animals. And you can also see there are differences in the different areas, but I'm not going to talk about that today. And based on these scans, we were able to model the so-called cerebral metabolic rate, which is the measurement of glucose consumption of the brain, which is glucose per weight per minute. Not that important. And we basically could find a CMR of 28 in the forebrain, 25 in the cerebellum, and approximately 26 in the whole brain, and lower in anesthetized animals. That is interesting, but more interesting is if you compare that to mammalian species. And we were actually really puzzled when we first saw the results of this comparison. And therefore we did all these control experiments. but it turned out that our results were correct, and yep, that we have to kind of live with that. Before I talk about pigeons, let's first have a brief look on the energy consumption of the typical mammalian neuron. So what we have plotted here is a CMR by neuron densities. And you can see that mammalian species all fall on one regression line. So that means that on average, a mammalian neuron, irrespective of species, requires the same amount of energy, which is approximately 5.8 times 10 to the minus 9s. What you can also see is that the pigeon does not really fall onto this regression line. It is much lower, and the absolute numbers for the pigeon are 1.78 times 10 up to the minus 9s, which means that pigeons have a CMR per neuron which is 3.25 times lower. That means a pigeon neuron needs much, much, much less energy than a mammalian neuron, and then can possibly explain how pigeons, and likely also other bird species, can afford so many neurons in their small brains. Currently, we do not have a final explanation why this is the case. That could be adaptations towards flight. That could have something to do with the size of the neurons of pigeons and other birds, which are in general smaller. It could have something to do with connectivity. But this is all speculation. What I can tell you is that the energy consumption of the pigeon brain is much lower than a mammalian species. And this is basically in a nutshell what I just told you, so I don't have to repeat that. What is also interesting, if you compare absolute numbers, you see that the glucose consumption per day is approximately 0.414 gram of the pigeon brain, which is half of that of the rat. And as I said, if you then take neural numbers into account, it's even lower with 3.25. And I'm now at the end of my talk. So what I hopefully could show you is that MRI, fMRI experiments are a feasible option in crocodilian species, or in reptilian species in general, but they have to be adapted to these species. I could show you that the auditory and visual areas in crocodiles have a similar layout to their avian counterparts, and I just showed you that the energy consumption of the avian brain, or at least the pigeon brain, is approximately 3 times lower, which could explain how these species can accumulate so many neurons in their brain. And this, on the other hand, could explain how at least some avian species could come up with amazing cognitive capabilities. And yeah, I'm at the end of my talk. I have to thank a couple of people, my collaborators, obviously, and my old lab in Bochum and my new lab in Düsseldorf. And I obviously also have to thank you. If you have any questions, I think we have now some time to discuss. And yeah, thanks for your attention.
Thank you so much for this fascinating talk. I sure learned a lot about 2 different species. I think we could probably just move straight into the general Q&A, so we have a bit more time with that. Yeah, let's go ahead and begin, because I'm sure there will be a lot of questions. So if you have questions for any 3 of the speakers from this session, so that includes Pascaline, Julien, and Felix, now we could address the 3 of them. So many fascinating things. I'm really humbled to be a mammal after learning about how we really declined in neuron density and sensory systems and in EQ. So it doesn't sound that fascinating to be a mammal anymore after this for me, but I'm sure fascinated by other species. So any questions to start us off? You could just type them in as well.
All right, so it's my pleasure to chair the second session and introduce our speakers. I'd just like to let everybody know that unfortunately Colette De Hay cancelled with us last night. She's had a personal emergency, so the talk from 1:30 until 2 is not going to take place. So I was thinking perhaps we could just have the three speakers just speak one after each other and then have a bit more time for Q&A and the coffee break. Is that okay with everybody? Or should we have that session in case somebody wants to say specifically come for Chet's talk? Anyone? No, just play, it's the three of them. All right, so then it's my absolute pleasure to introduce Prof. Paul Manger from the University of the Witwatersrand, and he'll be speaking on what are big brains and when have they evolved. Prof, are you here?
Hi, Tanya. Thanks for inviting me to the talk. Can everyone hear me okay?
Yes.
All right, now I need to share my screen, don't I?
Yes.
So there we go, share that. Is that visible? I have to hit slideshow, yeah?
Yes.
From start. Yeah, does that look good?
Yes.
All right, great. So what I want to talk about today, and it'll be a brief talk, is about big brains and when they've evolved. So what really are big brains? So in the literature, we hear a lot of talk about big brains and how amazing they are. And that's a very anthropocentric point of view. But we've taken that anthropocentric point of view and really lumped that on a bunch of different animals. And big brains are something that's talked about a lot. So I want to sort of try to bring some clarity into this discussion about what we mean, or what, sorry, I should say what I mean when I talk about big brains, and hopefully get people to agree with what I mean. So if we look across vertebrate classes, the biggest brains are found in mammals. And I don't know, can you see my pointer there? That's coming up, okay. So the biggest brains are found in mammals, and in the sperm whale and the killer whale, they can get up to around 9,000 grams. If we look at birds, reptiles, amphibians, and fish, they have much smaller brains, maxing out at about 200 grams for the manta ray, which is the biggest brain amongst the fish. And then if we look at amphibians, even in the largest fossil amphibians, the brains only get up to about 20 grams or so. So when we're talking about large brains, when we're talking specifically about large brains, we really can't talk about large brains in non-mammalian species. That to me just doesn't seem to work. You can talk about larger or smaller brains within a particular group, but not saying that these animals have large brains. That would be disingenuous. It wouldn't be really telling the whole truth. So if we're going to, so now I'm just going to focus on mammals. Now I've done away with the majority of vertebrates. I'll focus on the little group of mammals that we're part of. And if we look at brain size across mammals, we see that there's quite a range from very small brains down around 100 milligrams or so, up to the larger brains like here with the elephants, humans are sitting over here, and so on. And we're all familiar with these plots of body mass against brain mass. And the really interesting thing that comes out from this when you look closely at the data is that there are very strong regressions. If you look at mammals on their own, the probability that body mass and brain mass are not correlated is 10 to the minus 294. So that's an incredibly strong, I call it a structural law of form. We see primates in general have slightly larger brains than the other mammals. Again, the probability that this is not correlated amongst the primates is 10 to the minus 48. So these are very, very strong structural laws of form. Over here in the red, we have the hominids, and up here I have data from 2 elephants, the extant averages from the 2 extant elephant species, or the Indian elephant and the African elephant. What we see though is when we look at the other mammals, we start to see that around 700 grams, body size can increase dramatically. Remember, these are logarithmic scales, but brain size seems to be restricted to a level below 700 grams. There's something happening at this point at around 700 grams that makes it difficult for brains to get bigger than 700 grams. So, and I'll come back to that later in the talk, but it's this 700 gram ceiling on brain size that is only exceeded in a few cases that is really, really interesting. And I think that's a very interesting point. And so from now on, when I'm talking about big brains or large brains, I'm talking about brains that exceed 700 grams in mass. That's my personal specific definition of it. If we look at this, what I call the over 700 gram club, it's an idea I've floated around for a while. There's 7 lineages within this over 700 gram club. There's the majority of the genus Homo. There's the extant elephants, the majority of the species of extant whales and dolphins, 3 species of seals, and 1 camel species. Now, these are all sort of very phylogenetically and structurally distinct large brains. The brain of a seal is not the same as the brain of an elephant. There are many, many differences there. So there's several different trajectories or phylogenetic trajectories that have been taken to evolve a large brain. So I want to sort of run through these cases and point out what I think are the interesting features with this. So if we look at pinnipeds and the Bactrian camel first, these are sort of the more borderline cases in terms of being over 700 grams. So if we look at this regression on the left here for the pinnipeds, we have a beautiful, highly correlated regression for body mass against brain mass for most of the seals. We have confidence intervals and prediction intervals. And those seals that fall with brain masses above 700 grams, here we see the Stellus, no, it's not, I've forgotten what Murrawinga is. Oh, I think it's the elephant seal. Sorry, that's the elephant seal, the walrus, and the leopard seal. These are the 3 seals that have a larger than 700 gram brain. And if we look over here with the Bactrian camel, its brain is about 738 grams. So it just sneaks up over the 700 gram barrier. But both of these examples appear to be extensions of lineage-specific allometry, the relationship between body mass and brain mass. It would be hard to argue statistically that these are significantly different to the other members of their lineage and need to be separated out from them. So that's all I want to say about those guys. So in 4 of the species that have over 700 gram brains, it looks to be just an extension of allometry. So when I say 700 grams, there's a bit of a play around that as well. We go on, sorry, I'm just going on to my next slide here. I missed a slide. So let's talk now about human brain evolution. We've all seen how primates have a larger relative brain size than most other mammals. And then in the hominid lineage, there's a big difference here. This is Homo sapiens sitting here, Neanderthal, and there's some of the more recently discovered fossil hominids that I haven't included in here, like Naledi, but they fit to these allometries very nicely. We see amongst most primates, including the great apes up here, we have this beautiful allometry between body mass and brain mass. Then it changes dramatically in the hominid lineage. There's still a very strong, statistically significant relationship between body and brain mass. What's most interesting is the slope of the allometry in the primates is at 0.76, which is a negative allometry, meaning that for every doubling in body mass, brain mass increases by about 1.3 times. However, in the hominids, there's a positive allometry, so that for every doubling in body mass, brain mass increases by about 3 times in size. So there's something definitely unusual happening in the hominid lineage. And this doesn't like to move. It doesn't like to move forward. It takes 2 jumps and jumps back. So when is this happening? What do we see over time? And if we look on the bottom, on these graphs at the bottom here, we have millions of years before present, cranial capacity for the left-hand side graph, and body mass for the right-hand side graph. I've distinguished the Homo lineage from the Australopithecines. And what we see for many, many millions of years, or several million years, in the hominid lineage, cranial capacity and body mass in the Australopithecines stayed pretty much the same, very close to what we see in the extant great apes. It's quite similar in that sense. However, after the appearance of the Homo lineage, we see Homo habilis was very similar to the Australopithecines, and then there's a big jump up to Homo ergaster. And if we go back one slide, we can see here are the Australopithecines down here and Homo habilis, and then there's a big jump up to ergaster here. And this sort of creates this positive lineage, but it fits very nice statistically. So what we can see if we look at evolution of the brain in humans, brain mass in humans, for the most part, we can say that it's explained by structural laws of form or scaling with body mass. There's a possible adaptive event that occurred at the origin of hominids. My feeling is this is likely to be bipedalism. It may have been a positively selected change, which is an adaptation, or it could have been just a neutral change. And why I say a neutral change is that the Australopithecines were bipedal, and they didn't increase in brain mass and body mass until a few million years later. So these things, but this possibly neutral change in the relationship between brain mass and body mass may have caused major changes later in the evolutionary history of the hominids. So, but the important point I want to get across here is that there was a period of stasis in brain and body mass, followed by a period of increases in both. And we're still sort of in that period of increase. If we look at cetaceans now, and I think most people know I'm a bit infamous for challenging the ideas of big brains and cetaceans. If we look at allometry across mammals, we see mammals in green, the hominids in black here, cetaceans coming across here in pink. We have some animals that have relatively large brains, some animals that have relatively small brains, but what they have in common is they all have quite large brains. There's only a few species, only about a third of the species of cetaceans have brains smaller than 700 grams. So when did these guys evolve these large brains? If we look through the fossil record, we can see the archaic ungulate lineage. And this is a lineage it's mostly made up of artiodactyls, to which the cetaceans are related. You see the archaic ungulates, and across time, the relative brain size is slowly increasing up until we get to modern ungulates or modern artiodactyls. I think it includes parasidactyls as well. When we look at the ancestors of cetaceans, the first aquatic cetaceans that were in the water, these are the Archaeocidae, and their brain-body relationships appear to be quite similar to what we see in their land relatives. However, there's a jump between the Archaeocidae at the Oligocene Odontocetes and the Miocene Odontocetes and the extant whales and dolphins all appear to have very similar brain to body mass ratios. And statistically, they're quite similar. So there was a jump after that. It was about 20 million years after they were in the water, the Archaeocidae, that suddenly the larger brain of the Neoceti or the modern cetacean fauna appeared. So if we look at cetacean brain history, they were first adapted to an obligatory aquatic lifestyle around 50 million years ago. Their relative brain size and the scaling of it was comparable to their relatives that were living on land. Then 32 million years ago, there was a large change in brain size. And this change in brain size coincided with major environmental shifts. After that, brain sizes remained relatively stable since. So the important thing is we again see an extended period of stasis followed by a rapid enlargement. This is the second time we've seen this in animals that have brains greater than 700 grams. Why is this not? There we go. Okay, sorry, it's just moving too fast. It takes a while for the computer to catch up to my pushing of the buttons. So let's look at elephant brain mass. We know that elephants have brains that are around 5 kilograms, so they definitely exceed the 700-gram mass. This is an image taken from a paper that Julien Benoit got together, and I was lucky enough to work on it with him. What we see is that the ancient proboscideans, the pre-elephants, had quite small brains, around 400 grams for their body size, very small relative brain size. But around 35 million years ago, their brains exceeded the 700-gram limit and then became quite large. We're looking at brains that are around 3 kilograms around the early Oligocene. So they've increased in size, and it's often in relation to the body mass, to increases in body mass. There have been some occasions, and Julien showed that beautifully in this study, where there were increases in relative brain mass in the lineage in a couple of different places. If we look at, if we look at body mass in elephants, an interesting story comes out. And this is from a bunch of data I collected from the literature. At around 38 million years ago, we see a major jump in body mass. And we know that brain mass and body mass are very tightly tied together. We've seen in this case, around 38 million years ago, we're finding the larger brains in these animals, we're finding the larger bodies. And this is a third example where there was a long period of stasis prior to a major shift in both body mass and in brain mass. So now we've got 3 examples showing the same thing. So what are the similarities and differences in these lineages? So in the hominids, the elephants and the whales and dolphins, they all have species with brains larger than 700 grams. These are all independent phylogenetic events. They're not related to each other, and they've occurred at very different times. About 38 million years ago for the elephant lineage, 32 million years ago for the whale and dolphin lineage, and around 1.7 million years ago in the hominids. So the brains also of these different groups are very different in structure, both at macro and micro anatomical levels, going down to the neurochemistry that we've looked at so far. And these lineages evolve the large brains in very, very different environments. So there's sort of some similarities, but there's also a lot of differences. But let's focus on some of the similarities. Change slide. It's probably going to go through 2 changes now. Okay, so similarities in these lineages. All 3 have this extended period of time when brain mass and body mass are stable and comparable to closely related species. All 3 lineages underwent changes in both body mass and brain mass. Some of these can be tracked in an allometric fashion. For the proboscids or the elephant lineage, there was body size increases and a few relative brain size increases, but quite minor compared to some of the other species. This is when we can track the changes mostly with allometry, but there are exceptions, which Julien's shown very nicely. If we look at cetaceans, what's happened in their evolutionary history was there is a decrease in body size, but a slower decrease in brain size that's led to larger relative brain sizes. And this has evolved and changed throughout the course of their history. And in the hominids, there's the change to positive allometry, leading to larger bodies and rapidly increasing brain masses. But why do we have this sort of period of stasis followed by rapid change? So the kind of question I ask about this is, what triggered or allowed these changes to occur? Are there environmental pressures that drive this process? I think there are, but I think there's something more than just environmental processes. We know that the mammalian brain produces heat independently of the body. What I've proposed is that there is a neurothermogenic system within the mammalian brain. This neurothermogenic system basically relies on what's the word, non-shivering thermogenesis. It works in a very similar manner to the way heat is produced in brown fat. It's actually really, really quite similar. There is a system that occurs in the mammalian brain that produces heat. However, the capacity of this system has a limited scope. And this is why we end up getting that sort of 700-gram ceiling in brain mass across mammals. So in order to break through this ceiling, we need specializations of the neurothermogenic system. So what I'm thinking now is perhaps we need a combination of environmental pressures along with neurothermogenetic specializations in order to evolve large brains. I personally think the environmental pressures are actually acting on body mass, and that it's only when we evolve these neurothermogenic specializations can body mass and brain mass increase at the same time. We need two things to happen in order to evolve brains that can break through the 700-gram ceiling. So if we look at the cetaceans, and this is something we published earlier this year, at the time of the evolution of the large brains in the whales and dolphins, there was a major environmental cooling of oceanic temperatures. For a mammal living in the water, that is a really huge pressure. What we see in the brain of whales and dolphins is that there's specializations of both neurons and glia for thermogenesis. On the left here, we have a section from a minke whale brain. This is a Nissl stain in purple. Here is an immunostain for uncoupling protein 1. And basically we see that every neuron has uncoupling protein 1 in it. The uncoupling proteins are proteins that disrupt the mitochondrial metabolism and release all the energy that was going to go into the ATP cycle as heat. So they can allow for heat production. And if we look at the glia, we find in the cortex here, this is in a harbor porpoise. This is the cortex of the harbor porpoise stained with Nissl. We see about 30 to 35% of the glia stain up beautifully for uncoupling protein 4. And in the white matter, we see about 60% of the glia stain up for uncoupling protein 4. So we have two, well, very distinct specializations in both the neurons and the glia for thermogenesis in the cetaceans. So what about in hominids and proboscideans? And this is data I haven't published yet. We find that there are neurothermogenic specializations in both hominids and proscidians, well, in humans, but it only occurs in the white matter. So unlike the whales and dolphins, where there's a lot of specializations in the neurons and so on, that doesn't occur in the hominids and the elephants. But in the white matter, we see this is the glial Nissl stain for glia in the white matter of a human brain. Here we see this is uncoupling protein 3 staining up different glia, and you can actually see all the filopodia of the glia as well. Here's a slightly lower magnification picture image of the white matter stained for Nissl in the elephant. And here we have it stained for uncoupling protein 3 again. And we have about 60% of the glia is immunopositive for uncoupling protein. So we have, again, neurothermogenic specializations. However, in their evolutionary history, perhaps it was really important or advantageous for these lineages to have much larger bodies. It's quite different to the situation in whales and dolphins, where the cooler oceanic temperatures could drive those kind of things. Maybe in the paleo environment that these animals inhabited, having a larger body was advantageous. But it took them several million years to get to the point where the brain caught up and said, aha, if I have this neurothermogenic specialization, you can evolve that larger body, inhabit that new niche, and I come along for the ride and I get a bigger brain as well with it. So in summary, the way that I'm thinking at the moment, and obviously I stand to be corrected on all these things, these are just ideas. large brains should only be used to refer to brains that are greater than 700 grams in mass. Anything that's smaller than this can be primarily explained by allometry. And we don't need to have adaptive rationale for brain size in the vast majority of vertebrate species. Truly distinct large brains really only appeared in 3 lineages: the hominids, the proboscideans, and the cetaceans. It seems to me that there were environmental pressures on the animals that where a larger body mass would be advantageous, but they couldn't evolve a larger body mass until there was a specialization of the neurothermogenetic system within the brains of the animals. And when that happened, larger bodies and larger brains could evolve. The last thing I want to point out is that having a large brain doesn't necessarily equal intelligence. That's one of the biggest problems we have, I think, in the comparative literature, is a big brain equals intelligence. We need both the appropriate internal structure and the large brain to produce brains that can really do the kind of things that we're doing now, like talking about large brain evolution across different countries over the internet and really trying to compare our ideas. So I think this is a really important sort of aspect to think about. And we need to break away from what I think is probably a false assumption that large brains equal intelligence. And that's where I want to end. Thank you.
Thank you so much, Prof. That was really good. We've got questions and answers at the end. So I think we're just going to move on to Philipp Gunz. He's talking on connecting genotype and phenotype, the next frontier in the palaeosciences.
Need to connect my camera. Okay, I think this works, I hope. Then let me share my screen. Give me 1 second. Share screen. I presume this works, right?
Yes.
Okay, so this is, thanks a lot for having me. I'm really honored by the invitation. I wish we could do this in person, but okay, now we're here in front of our screens. So I'll talk about connecting genotype and phenotype, and I'll mostly talk about the fact that you all have weird heads. And the reason I know that is because all modern humans have very unusual head shapes, and I want to talk about the biological processes that lead to this and what they may tell us about the evolution of our own brain. And I'm a group leader at the Max Planck Institute for Evolutionary Anthropology in Leipzig. And yeah, so this is what this looks like in a pandemic. Usually there are more people around here. And the main theme of my research is trying to understand how evolutionary changes of development shape organisms. And I want to basically reverse engineer in a sequence of pictures the ontogenetic differences that lead to the shape differences that we see in bonobos on the left-hand side and modern humans on the right-hand side. So as adults, they look very different. I think this is no surprise to you. But as we go back in development, so this would be around the time of birth, and this is these are micro CT scans of fetal individuals. And I think it's easy to appreciate that they're not exactly the same, but that they look much more similar than these two groups look as adults. And so there are very similar starting shapes prenatally that develop differently along different ontogenetic trajectories. And we know from genetic data that these ontogenetic differences are largely driven by differences in the regulation of gene expression, right? Rather than single point mutations or I mean, there are some, but they mostly affect the expression of genes that exist in both species. And of course, to focus on the main topic of this workshop, we have to accommodate the inconvenient absence of fossil brains. And I'm not going to talk much about this particular study, but just to illustrate that, as was mentioned already quite some time in talks this morning, there are ways to look at the interior molds of the brain case, so-called endocasts, and get some information about, brain, or let's say endocranial volume and organization and structure of the brain. But of course, we can't look inside. And the good thing about hominins is that we know that early brain growth is really the main driving factor that shapes the brain case. So we do, by looking at these endocasts, we do have a very clear picture of at least what the outer size of, outer structure of the brain and its surrounding structures look like. So here, this is an ontogenetic sequence in modern humans between 14 weeks after gestation until 24 weeks. And you can really see that early in fetal development and throughout the first year of life, it's really the brain case is very, very thin. The sutures are wide open, and basically it's the growing brain and the surrounding fluid that push out the brain case. And this is why we can study brain evolution even if we don't have brains preserved in the fossil record. But as my title promised, the main theme today was trying to connect genotype and phenotype. And of course, there are two big challenges. And the one challenge is that most, if not all, of the traits that we are interested in are complex traits. So they're not simple Mendelian traits. They're influenced by many genes. And as I mentioned before, that when we look about changes in the hominid or the hominin lineage in particular, we're largely talking about evolutionary changes of gene expression, which are trickier to assess than single point mutations or so on. And I want to focus, as promised, on the difference between modern humans on the right-hand side and our closest extinct relative, the Neanderthal, shown here on the left-hand side. And I think most of you know that on average, Neanderthals and modern humans had very similar endocranial volumes. On average, actually, Neanderthals had slightly higher endocranial volumes, but basically the ranges overlap. But they have different shapes. And it's basically the modern human head that is unusual. We have these globular endocranial shapes that differ from every other hominin. And the reason why we have globular endocranial shapes is because we have globular brains. And the differences are most pronounced in basically the parietal area and in the posterior cranial fossa that houses the cerebellum. And what's interesting is that we are not born with globular brains. So this is a modern human neonate, CT scanned on the 7th day after birth for some other medical issue. And if you were to scale up this modern human baby to the size of an adult Neanderthal, you would see, or you can see here that, okay, it still has a baby face and no teeth, but the endocranial shapes are actually very similar. So all modern human neonates have very elongated endocrania. So my group did a while ago is trying to understand, okay, are the differences in the developmental patterns of the endocranium that, explain aspects of this modern human globular phenotype. So we looked at Neanderthal babies and how they change their shape throughout development. And what we found is that Neanderthals have Neanderthal-like faces already at the time of birth. So they have bigger noses and differently shaped orbits. And there are also features in the cranial base that are clearly Neanderthal-like. But if you superimpose these two poster children here, that's the same neonate that I showed before, shown semi-transparently in blue here on the right-hand side, and the Neanderthal child endocasts in red. You can see that they have very similar endocranial volumes and very, very similar endocranial shapes. And this implies that the pronounced shape difference that we see in endocranial shapes between Neanderthal adults and modern human adults develops largely after birth. And here you can see this. So in basically between birth and the eruption of the deciduous dentition, so we're talking basically about a process that happens in the first year of life, we not only see a change in endocranial volume in modern humans, but we also see a change in endocranial shape. And it's most pronounced in the parietal area and in the cerebellar area. So the reason why this is interesting and may tell us something about the mind is not because I want to argue that having a round head is in any way advantageous or was a primary target of selection. What I'm trying to say is, okay, this tells us something about the time window in which this phenotype develops, right? And the time window between birth and say the first year of life is particularly important for establishing the network connectivity inside the brain and myelination, right? So it's not neurogenesis. It may have to do with myelination. And so we know when this happens during development. We also have an idea about the evolutionary trajectory within the human lineage, the Homo sapiens lineage, because the earliest representatives of Homo sapiens dated to around 300,000 years from Jebel Yehud in Morocco. They are endocranial volume-wise, they fall right within modern human variation, but endocranial shape-wise, they look very archaic. So they have a very elongated brain still. And as you move up in time within the Homo sapiens lineage, we see globularization, maybe even a gradual process, but the fossil record is too scarce to really say something about the exact nature of the process. And what's interesting here is that we see two informations on the right-hand side. We see the areas that change most in shape, and also color-coded the areas of the endocast that also change in size. And the green coloring in the cerebellar area here means that this cerebellar bulging actually corresponds to an increase in surface area of that region of the brain. But note that the parietal bulging that is an essential part of the globularization phase is white. And that means that it stays the same in relative size. And that to me means that something inside the brain is actually pushing out the parietals, right? There's a subcortical volume that increases and pushes out the parietal area. And here's the back and forth. So that's the average of early Homo sapiens versus the average of modern humans, elongated versus globular. And as I mentioned before, we have this sequence. So we also know that something similar happens during ontogeny. So I'm not sure how well the movie translates, but these are the shape changes in the first year of life during this globularization phase. And here, Simon Neubauer color-coded slightly differently than in the previous image, but red is really high increase in relative size, and white means no increase. And again, the cerebellar bulging is an actual increase in size and volume, whereas the parietal area stays white as it bulges, so something else must push out the parietals. So what's happening? And to answer that question, my colleagues and I looked at the combination of genetics and brain imaging, and we took advantage of the fact that modern people outside of Africa carry the genetic signature of interbreeding events with Neanderthals inside their genome. So all of us with non-African ancestry carry around 1 to 2% of Neanderthal genes in us. And because these genes are maybe not randomly, but scattered across the genome, and your Neanderthal genes differ from mine, we can collectively reconstruct around 40% of the Neanderthal genome from modern day people. So what we did is we combined paleoanthropological data from the fossils with neuroimaging and genomics and gene expression data. And then, because we're really looking for the needle in a haystack, so what we targeted our search on is the phenotypic and genetic differences between Neanderthals and modern humans. And the idea basically was to constrain the search space of our study. And the simple question was, are there any Neanderthal introgressed fragments in modern Europeans linked to a less globular endocranial shape? And here is an example of, on the left-hand side, a modern human MRI scan with a elongated shape, and a modern human with a very globular shape on the right-hand side. So there is variation within modern humans, and we wanted to see if there are any Neanderthal fragments that are associated with that variation. And what we did is basically scale up our phenotyping approach that we've developed initially for endocasts and fossils to include MRI data of around 4,500 modern Europeans from Germany and the Netherlands. And here on the left-hand side, you see the endocranial shape differences between Homo erectus in green, Neanderthals in red, and modern humans in blue. And you see that they're quite distinct. And if we basically then project the 4,500 modern human MRI scans into that space, we see a nice overlap between the CT and the MRI data. And we see that there's a lot of variability, even just among recent Europeans. I then try to simplify that multivariate space by computing a simple metric. So we basically end up with one number that tells us how round or how elongated your brain is. And we call this the globularity score. And it's basically just the mean difference between Neanderthals and the mean difference between Neanderthals and modern humans. That's a multivariate vector. And then we project each individual onto that vector, and then we end up with one single number. And then we looked for association of this globularity score with 50,000 archaic SNPs, single nucleotide polymorphisms. And the way to read this plot, these are the p-values, is basically where they fall on or very close to the line, the expected line, the red line. This is just noise. This is just statistical noise. But the points up here in the right corner, these are significantly associated SNPs, well, significantly associated with the globularity score. And we looked at this. And once you correct for multiple testing with the p-values, then we are left with two significant SNPs. And they have a subtle effect, and they all make the, well, they have the same effect in 5 different data sets. And one is on chromosome 1 and the other one on chromosome 18. And they both push the endocranial shape or the brain shape towards a slightly more elongated shape, so more Neanderthal-like if you want. And what's interesting is that both SNPs, or significant SNPs, they fall into the non-coding region of the genome, right? So they don't change what is being expressed, but what they do change is how much is being expressed and where. So the significant hit on chromosome 1 changes the gene expression of a gene called UBR4 in the putamen, which is a part of the basal ganglia in the brain, a subcortical structure. And we know what this gene does, at least we know it experimentally in mice. So the mouse version of this gene is called P600, and we know from knockout studies that it regulates neurogenesis. And so if you basically lose this gene entirely, or if you knock it out, then you have a microcephalic phenotype in mice. And the other significant hit was on chromosome 18. Initially, I thought it was called Philipp, but sadly it's pronounced Flip. And what it does is it affects the gene expression of FLIP1 in the cerebellum. So heterozygous carriers of that gene, so if you have a Neanderthal allele from your mother or father, then you have more FLIP1 expression in the cerebellum. And we also know from mouse studies what FLIP1 does, because it encodes a negative regulator of a key signaling pathway that drives myelination. So it's the AKT signaling pathway. And so basically, if you overexpress this in transgenic mice, you have hypermyelination compared to the wild type controls. And because, as I briefly mentioned before, in carriers of the Neanderthal allele, this FLIP1 expression is slightly higher in the cerebellum. This predicts a dampening effect on cerebellar myelination via that pathway in Neanderthal carriers. Again, I mean, most of these effects are fairly subtle, but I think it's very, very interesting that they show up in the cerebellum and in the putamen, because these are classic motor cortex regions, right? I mean, well, let's put it that way. They're not motor cortex, but they receive direct input from the motor cortex, and they're involved in the preparation and learning and coordination of movements. And the basal ganglia, they also contribute to higher cognitive functions and are important for memory and attention and planning and also skill learning. And they have also been implicated in speech and language evolution. So basically these genetic hints, these echoes from our interbreeding events with Neanderthals, they may shed light on some of the biological pathways that are important for the evolution of this uniquely globular phenotype. I want to give you a sneak peek of what we can do to push this further, because my colleagues in the Netherlands and I have been working on a genome-wide association study. So not just looking at the Neanderthal fragments, but basically looking at all the genome using the UK Biobank data. And the UK Biobank data is just amazing because it allowed us to basically scale that study to more than 33,000 participants, almost 34,000 participants. And what we found, and this is the work by Barbara Moltz in Simon Fischer's lab in Nijmegen, using genome-wide association studies, is that there are around 200 genes that are likely involved that are associated with this globularity score, and 27 associated genetic loci. And what's interesting here is that some of the top hits include the ventricular system and distinct white matter tracts involved in determining brain shape. And so the white matter, I think, okay, it's another hour of talking about these tracts in particular, but I want to briefly mention that okay, basically the volume of the ventricles and also is affected by it's also linked to the globularity score. And I think what this means is that there is like aspects of globularity are driven by, I would guess, an increase in fluid and fluid production inside the ventricles. So basically, aspects of it are purely mechanical, I guess, during autogeny, but other aspects are, for example, linked to white matter. So to, sum up, I want to give basically rehashed the idea that I think what is important is that we can use known phenotypic differences of closely related species to constrain the search space, and also take advantage of interbreeding events between hominin groups, because otherwise I think it's a next to impossible task to find needles in the haystack, unless you can really work with data like the UK Biobank, where you have ultimately there will be like 100,000 participants. So then you probably don't need to constrain the search space that much. But when we constrain the search space, we can basically also look at other closely related species, right? We can look at the difference between bonobos and chimpanzees, and basically scale this up to include other hominids, gorillas, and orangutans, and also the different subspecies. And what my colleagues in Leipzig and I are trying to do as part of a large network of collaborators is basically trying to understand the evolution of skulls and brains in our closely related hominid cousins. So what we are basically trying to reimagine is how to do ethical studies of brains in non-human primates. And what my colleagues in primatology are doing is basically they're observing primates in the wild, and when they die from natural causes, a veterinarian on site goes there, cuts up the skull, and extracts the brain, puts in formaldehyde, and then ships it to Leipzig to have it MRI and CT scanned. And here you see, a first batch of data. So we have the bones and the white matter tracts and the endocast and the gray matter information of the same individual. And what we want to do basically is combine these different sources of information and also connect it with the genotype, because of course, if we have all the soft tissue, we can easily sequence them. So with that, I want to thank you again for having me. I want to thank you for your attention, and I'm happy to discuss any issues that may arise in the next section.
Thank you so much, Philip. I'm sure there's going to be a few questions in the Q&A session. But so next up, it's Chet Sherwood, and he's going to be talking about human neurobiology in comparative perspective.
Hello, everyone. I am really happy to have the opportunity to participate, and want to thank the organizers who put together a really fantastic program across continents and across paleoanthropology and comparative neuroanatomy. It's something I'm very enthusiastic about, and so happy to be here. Also, I think even though unfortunately we can't be all together, I got a little bit of a taste of the international travel experience, because I had to wake up at about 4 in the morning to get my way to the office where it's quiet from the children who would otherwise be waking up and getting off to school. So it's almost 7 in the morning here in Washington, DC. And so I've had 1 cup of coffee and I think I'm ready to go. So I'm here to talk about human neurobiology in comparative perspective. And in this brief talk, I'll be giving an overview of research findings from our lab for evolutionary neuroscience at the George Washington University, in collaboration with other laboratories, all towards the aim of trying to place human brain structure in a comparative evolutionary context. And Paul's talk earlier referenced other large brain species, and I think that comparative context is really important to recognize. Humans are not the only ones that have large brains, relatively slow development, high degree of sociality, and an extended lifespan. And really wonderful examples include, of course, odontocetes and elephants, and researchers like Paul and others are working to try and understand their neurobiology even better. Closer to home for us on the tree of life are the other primates. And of course, they're relevant to understanding human neurobiology because of their phylogenetic proximity and various similarities in biology. And also, it's a great opportunity to study diversity. There are, depending on how you do your taxonomy, upwards of 350 different species of primates living today that show a really terrific range of body size, ecological adaptation, social behavior, and really provide great opportunities for comparative analyses to put humans in context. So this is something that this crowd is definitely familiar with. When you do compare human brains to those of other primates, there are some things that are immediately quite conspicuous. Human brains are large in size due to a large number of neurons, and also the white matter, which represents the connections among neurons. Human brains are about 3 times larger than those of our closest living relatives, chimpanzees and other great apes, and are also much larger than you'd expect for our body size. This means that a lot of what's observed in human neurobiology is likely correlated with this important fact of brain enlargement. And so the study of allometric scaling, which I'll refer to at several points in the presentation, does attempt to try to take this into account. So you can see most of human brain enlargement is represented by the cerebral cortex, and within the cerebral cortex, there's differential expansion of particular neocortical areas that are called association areas. These are areas that are in the prefrontal cortex, parietal and temporal lobe. They're important in humans for functions such as language and social cognition, long-term planning, abstract thinking, and so forth. So a fascinating region that includes association cortex is the insula. And the insula is buried underneath the overhanging frontal, temporal, and parietal lobes. There's an anterior posterior gradient in structure and function in the insula that is also represented in changes in cellular architecture of the neocortex in granularity, or the conspicuousness of the layer 4. And so the posterior insula displays activation. It's very closely adjacent to secondary somatosensory cortex, so its function is relatively similar, and it's activated in thermosensory information, processing of pain, hunger, thirst, and touch. But as you move more anteriorly within the insula, the anterior agranular regions are involved in functions like the ability to process subjective feelings and social awareness. So for example, one way of sort of thinking about this is that sensation of pain in one's own body activates posterior insula, whereas feeling empathy for another person's pain activates the anterior insula. And there are models that suggest that anterior insula is especially important in guiding social interactions and empathic understanding of other individuals. And indeed, diseases like frontotemporal dementia, which selectively destroy the anterior insular cortex, do severely impair social understanding. So we undertook a study some years ago to examine sizes of these portions of insular cortex, given their really interesting functions, potentially in the evolution of social cognition in primates. So what you can see here are representative coronal sections from various different primate species, going from more anterior to more posterior slices, where you can identify, based on that granularity of layer 4, these different subdivisions of insular cortex. And then you can measure their volumes and map them in these different species, which is what we did. Notably, it's interesting that in the anterior agranular insular cortex, in some primates, particularly humans and great apes, there's an even more distinctive subdivision because of the occurrence of high numbers of what are called phonoconomo neurons, which are specialized projection neurons in lower layer 5. And this is a topic of interest for a lot of researchers to try to understand the distribution and function of phonoconomo neurons. They're also selectively lost. They die in that disease, behavioral variant frontotemporal dementia, which affects the patient's sense of empathy and understanding. So in this study of evolution of insular cortex across primates, we were able to examine histological brain sections from 30 different species, individuals that had lived in US zoos and myobenical facilities. And there's a lot in this image, I acknowledge that. It'll be summarized in the next one, but this shows the overall scaling of the insula and its various subdivisions compared to brain size. There is a non-phylogenetic regression as well as PGLS lines, which are plotted, and they're very close to each other. They're basically overlapping, which shows that there's not very strong phylogenetic signal in this data set. But to summarize what you find, there is overall positive allometry of insular cortex compared to brain size. So as brains get bigger, disproportionately more and more of cortex is made up by insular regions. And then within those insular cortex subdivisions, you can also observe changes in allometric scaling. So as you go from the most posterior to the more anterior subdivisions of insular cortex, you have increasingly positive allometric scaling. And when you plot human values on the primate regression trends, you see that humans are not exceptional in the size of any of the subdivisions of insular cortex. We have a large anterior insular cortex, which we do employ in our very sophisticated social cognition, but it is not any larger than one would predict for a primate of our brain size scaling. And I would suggest that this might be true of other association cortex regions as well. In general, they are proportionally large, as you saw in that very narrow comparison of that human brain to the chimpanzee brain. But when you have a very broad cross-primate sample, perhaps they're not always so unexpectedly large. There's a lot of active, vigorous debate right now about whether that's the case for prefrontal cortex, but it does seem that quite a lot of this disproportionate enlargement of association regions comes along with very predictable cortical scaling. Another question that one can ask with these sorts of data is whether species variation in anterior agranular insular cortex might be associated with social cognition capacity across primates. So to examine this question, we used PGLS multiple regression of agranular anterior insular cortex volume compared to group size across primates. When you control for body size, there is a significant relationship, but once you factor in overall brain size in that predictable allometric scaling, there's no longer a significant relationship. Combining just these data from insular cortex regions and now looking at other cortical areas that have been measured similarly in humans and our close living relatives, chimpanzees and bonobos, from cytoarchitecture. There are lots of other ways to measure differential cortical expansions, but this is just a compilation in a table of data sets that have used histologically defined cytoarchitectonic boundaries to make measurements of cortical areas in humans and chimpanzees or bonobos. And you can see that, interestingly, in terms of fold change enlargement, that actually the areas of anterior agranular insular cortex are among the most differentially enlarged in humans, beyond what you would observe for overall neocortex size, which is about 4 times larger. Something else that I'll point out, which is really notable, is that these human compared to chimpanzee or human compared to bonobo fold changes in cortical areas can be very different. So, humans seem to have this differentially enlarged agranular insular cortex compared to chimpanzees, but not compared to bonobos. And if you were to observe the overall regression, it's because chimpanzees have a unexpectedly small insular cortex. And of course, there are differences in brain organization between chimpanzees and bonobos and in brain shape, as Philip referred to. There are differences in the connectivity of insular cortex to anterior cingulate cortex, as have been reported by other research groups, differences in amygdala organization and serotonergic innervation, as we've studied. And this really just highlights that chimpanzees on their own really should not be taken as a stand-in for the common ancestor from which hominins evolved. There are important species-specific adaptations within the chimpanzee lineage. The cortical association areas are populated by neurons in humans that are highly branched and integrate many, many synapses. And when you compare human pyramidal neurons for their morphology in something like a Golgi impregnation, like you see here, to those in chimpanzees, and count up the extent of dendritic branching and synaptic spine densities, you see that human pyramidal neurons have 3 times more dendritic branching than in chimpanzees, and also more synaptic spines. So we sought to put this sort of observation in a broader comparative context by examining the scaling of synapses relative to neurons in a more systematic manner across primates. We selected regions of interest in the primary visual cortex and in the inferior temporal cortex. The inferior temporal cortex is one of the highest order regions within the ventral visual stream, which is involved in visual object recognition. And within these regions of interest, both a primary and a higher order association cortex, in vision, we examined upper layer cortical, the upper layers of the cortex, as well as the lower layers of the cortex, because they house neurons that have very different canonical patterns of connectivity. Upper layers have neurons that mostly make cortical association projections, and lower layer neurons mostly make subcortical projections. We used immunohistochemistry against synaptophysin, which is a pre-synaptic vesicle associated protein, to label synapses and observe them under high magnification. You can see them, they appear almost like grains of sand under the microscope between the unstained neurons in this preparation. And then we use Nissl staining for labeling neurons, which can be visualized easily across these species, and quantified the variables using stereologic optical dissector methods. For this study, we were able to examine 25 species of primates across the clade. And so this is just raw plots of the values. Here, this is for synapse density. And this shows that synapse density is actually relatively invariant across these primates and these regions, and doesn't show very distinctive phylogenetic patterning. Across all these regions and species, there's only about a twofold variation in synaptic density. You can see the human value. I'm sorry, it's not that easy to make out, but it's towards the middle of this plot. And those values are among the highest for overall synaptic density. We validated the synaptic counts from immunohistochemistry also with electron microscopy in a subsample as well. And you can examine under electron microscopy directly the localization of synapses. And then you can see that in this subsample that as well, humans don't show really an extraordinary high density of synapses from this sample of primary visual cortex. Then moving on, we quantified neuron density, which shows much more pronounced species differences than the synaptic densities. And there's as much as a 6-fold difference in the sample for neuron densities. When you combine the relatively invariant synaptic density with the degree of variation in neuron density to calculate a value of synapses per neuron, here it's evident that humans have the largest value, with great apes next. And you can see that depicted with the warmer colors and the larger circles. So when you take these data and put them in the context of brain size scaling, you can use phylogenetic generalized least squares regression like we do here, and just taking those data for synapse density regressed against brain size. You see, again, it's invariant across phylogeny and to brain size. And humans with our very large brains might show moderately increased synaptic density based on our counts in our sample, but essentially within the range of what you'd expect. Neuron density, we demonstrate here with this sample what's been shown in other studies, which is a negative correlation. Brains get bigger, neurons become more spaced out within the cerebral cortex because they have more space taken up by dendritic branching and other microstructure. And humans are here, as expected, with neuron density that would be predicted for our brain size for these regions. Notably, the synapse density and neuron densities within the sample are uncorrelated with each other. So then moving on, the synapse per neuron ratio as it relates to brain size. And here you have a steady increase with brain size, which is significant, and humans have the highest values with the greatest number of synapses per neurons. So this sort of data, which takes a much more broad comparative perspective, together with other studies, show that the human cortex contains neurons that have more dendritic branching, relatively more synapses, are distributed more in upper layers, mature later, and make expanded long-range association connections. And all these features contribute to make human cortical neurons perhaps more integrative and have a greater capacity for plasticity. These features, especially the developmental trajectory it takes to achieve these large sizes and large number of synapses, also might make human neurons more vulnerable to neurodevelopmental diseases like schizophrenia, bipolar disorder, and depression. Another further important piece of the puzzle of human brain specialization also concerns how our brains process information in a manner that may be shifted to be more impacted by social reward signals. And this can be very important in shaping the direction of plasticity that our brains have to be responsive to social groups in culture. So in collaboration with Nenad Cheshton's lab some years ago, we used a combination of RNA-seq to measure whole transcriptome and immunohistochemistry to label cells in a broad survey of 16 brain regions from humans, chimpanzees, and macaques. And as you can see in the box plots here above the red arrow, among all the regions and transcripts that were analyzed, one of the major standouts was in the striatum, which showed significantly higher levels in humans of expression of the gene transcript for tyrosine hydroxylase, TH, and also DDC expression. And these are important enzymes and pathways that produce dopamine and serotonin. And then moving on to counts of actual cells within regions of the striatum, the caudate and the putamen, you can see that the percent of TH containing interneurons in the striatum are also selectively elevated in humans compared to other primates. In a recent follow-up extended allometric analysis that was led by Marianne Reganti's lab, we show that humans have significantly more tyrosine hydroxylase expressing axons, which are involved with dopamine expression in regions of the ventral striatum. So here showing in the nucleus accumbens and the human values, which are significantly greater based on phylogenetic and COVA tests. And then also in the ventral pallidum, which is also greater on this analysis. So taken together, this might suggest that increased dopaminergic activation within the striatum, and particularly the ventral striatum, may be involved with enhancing social reinforcement reward in humans, which facilitates conformity, vocal motor learning, and so forth. And these human-specific brain evolutionary changes, we might hypothesize to be important in our species' cultural capacity. Okay, so for the final part of my presentation today, I want to spend a little time to talk about the National Chimpanzee Brain Resource. It's an open science project that was initially supported by the National Institutes of Health, and now it's carried on with additional support from the NIH and NSF on various grants. It was founded in collaboration between me, and Bill Hopkins, and Todd Proys, and currently Bill Hopkins and I are co-directors. It was started in 2015. So you can see the landing page for the website portal here, and what is it, and how can you use it potentially? It's a biorepository of fixed and frozen brains, as well as neuroimaging resources that include MRI data sets, both in vivo and postmortem, diffusion tensor imaging data sets, and then also associated records from many of the individual brains that are represented in the sample. So including pedigree, rearing history information, health records, individual performance on behavioral and cognitive tasks. Also part of it with a number of collaborations, we've been building up an RNA-seq resource of gene transcription in various brain regions from individuals in the sample. And we've also built and shared multiple segmented MRI atlases through the resource, as well as digital histology collections, which are consolidated. So altogether, this resource allows scientists who want to access it to perform studies that integrate psychological assessments with brain MRI scans, genotyping. Oh, and I should note that there's a very exciting initiative underway now to get whole genomes for hundreds of the chimpanzees in this database. And so that will be coming online, hopefully, within the next year or so. And I think that the NCBR has been pretty successful so far. Since 2015, when it opened up, we have fulfilled 112 requests from a wide range of national and international institutions, including probably some people at this conference. And it has yielded 99 publications related to the resource up to this point. And that is, to me, very exciting and very encouraging. And this is exactly what this is meant to do. And I would urge folks to explore the resource and find ways to do science with it. So how it's potentially relevant to also interpreting fossil hominins, it provides an opportunity to study variation in a species, chimpanzees, with large numbers of subjects that may be informative for interpreting fossil hominins. Of course, in human evolutionary history, before about 2 million years ago, cranial capacity is in the range of what you might see in modern chimpanzees or other great apes, until it started to increase more steadily up to this point today. But even more recently, there are other small-brained hominins like Homo floresiensis from the Indonesian island of Flores, Homo naledi, of course, from the Rising Star Caves in South Africa. And so being able to study large numbers of ape brains that are around this same size to explore variation, I think is really essential for interpreting these hominin endocasts. So just one example of how the NCBR's integrated data sets can be utilized comes from some of our own research. And this is one study that we did some years ago where we used an anatomical analytical method called source-based morphometry to investigate the association between individual variability in tool use skill in chimpanzees, where they use a probe to simulate termite fishing in a sample of 204 individuals in relation to their brain morphology from MRI scanning. And as you can see, the component from the source-based morphometry analysis that had the strongest relationship to tool use skill involved morphological variation in the inferior parietal and precuneus cortex. And what's interesting is that both of these areas are considered really hot spots of hominin brain evolution, especially potentially in the earlier Australopithecines based on sulcal configurations. And so this evidence strengthens the behavioral and paleoneurological connection with the stone tool evidence, potentially. Another way in which open data sets from chimpanzee brains have been used in paleoanthropology has been to provide a reference model for the localization of histologically mapped areas, such as Broca's area homologue. As we did in this study, based on 12 chimpanzee brains, and then registering them to a common template space for chimpanzees. And these data and other publicly available NCBR MRI scans can assist paleoanthropologists in analyzing fossil endocasts. So to wrap up, I think that understanding primate brain variation is really an essential context for interpreting the evolution of human neurobiology. You'll see that in many ways, once you take into account brain size enlargement, a lot of what flows from there is rather predictable, very significant for function and the evolution of our cognition, but can be understood in relation in this broader comparative context. And then also that comparative neuroanatomy and paleoneurology are complementary approaches that really have much to offer each other. And a conference like this, I think, is a really exciting opportunity to try to bring them closer and closer together and find fruitful ways to collaborate. So finally, I have to thank all the collaborators and funders that make this research possible. And I appreciate you all for inviting me and for your attention.
Thank you, Chet. That was a really excellent talk. I'm sure there's a lot of questions, and for those of you who came late, Collector Hay had to cancel with us last night due to some family issues. So the Q&A session will start now. There were only 3 speakers in this session. So does anybody want to open the Q&A session? I think you muted if you're talking, Sean.
Can you hear me now?
Yes.
Okay. Chet, I have a question for you. Before I do that, I want to point out that while you were showing the endocast of DH3, you were showing the skull of LES1, which is another Naledi, but a different Naledi, and it has a brain size of around 600 cc's. So anyway, just don't get confused, anyone.
Thanks for the clarification.
Yeah, I appreciate you showing it though. We're submitting that paper really soon. So you showed some data on the anterior insula and areas 44 and 45, and then later on talked about your own research on areas 44 and 45. But given its association with both the anterior insula and areas 44 and 45, and with empathy, have you done any work? Are you currently doing any work on area 47?
Sadly, no. That could be an interesting topic, but we are not. The answer is no. We are not currently doing any research on it. Yeah. Want to?
Yeah.
Yes. I mean, the resources are there, and really open to any opportunities to push forward research.
Does anyone else have a question? Feel free to raise your hand. Or Shannon, do you have a question?
Yes, thank you. My question goes to Philipp. First off, thank you so much for the presentation. I am really, really inspired. I've been really interested in a comparative approach between early modern humans and Neanderthals. So seeing your work was really remarkable. My question concerns the composite score that you spoke about. I think the globularity score. I think that there was a paper a bit earlier that was published and kind of had a Neander score, which was a composite genotype, and there were some issues around what that composite score really meant. And I'm wondering, with that phenotype composite score now, is there any concern that we should be having around it?
So thanks for the question. So yeah, it's an important difference, right? Whether you summarize the genotype or summarize the phenotype. What we did was summarize the phenotype, and I'm sure there are issues with it. Let me briefly explain to the others who probably have not read the other paper. Is what these authors did was something, I would say, comparable, but what they did is basically use the percentage of the Neanderthal genes, how many you have, right, as a predictor. And I think that's problematic because the amount of Neanderthal genes doesn't really tell you much, right? Because you could have the exact same amount, say, Chet and I, probably, let's assume we both have 2% Neanderthal genes in us. I guarantee you, we don't have the exact same Neanderthal fragments. So basically, I think this makes the genotype-phenotype connection impossible, is my critique of that approach. And of course, what you're pointing out is that we're also making some very simplifying assumptions when we summarize the difference between a Neanderthal and a modern human in a single number. And the reason is that you could you could have the same number by having, say, a very bulging parietal or a very bulging cerebellum, or a combination thereof. So it's not perfect, and the only way to do this better, I think, is to get better at automatic phenotyping, and there are methods that make this possible now, and scaling it up to larger data sets where we can handle much more complicated phenotypes. So to me, the study that I presented is just the tip of the iceberg, right? To me, it's a blueprint for other studies, but more, it's a proof of concept in my mind, not the final answer.